Have you ever stopped to listen to the quiet rhythm of your own breathing? Or felt the steady, silent pulse in your wrist? Right now, in this very moment of stillness, there is an entire universe within you, teeming with life and activity that never sleeps. You are a collection of trillions of tiny, industrious worlds, each one working in perfect harmony to allow you to think, to dream, and to rest. Tonight, we aren't just studying science, we are exploring the very architecture of your being, peeling back the layers of the mundane to reveal the extraordinary clockwork of the living cell. Welcome to this space of learning and quietude. I am so glad you've chosen to spend these hours with me. As we embark on this journey through the chemical foundations of biochemistry, I want you to let go of any pressure to perform or memorize. This isn't a classroom in the traditional sense. It is a sanctuary for your mind. If you find your thoughts drifting, let them go. If you feel sleep pulling at the edges of your consciousness, surrender to it. Your subconscious is a vast and capable ocean, and it will gather the pearls of wisdom we discuss even as you drift into dreams. Before we begin our deep dive into the cellular foundations of life, if you find value in these quiet explorations, I invite you to subscribe to our channel and join our community of learners. Your presence here is a gift, and I would love to continue this journey with you. Now let us begin. Lesson 1. The cellular foundations of life. In our journey to understand the vast and intricate world of biochemistry, we must begin at the very beginning, the stage upon which the entire drama of life is performed. We call this stage the cell. In our previous overview, we touched upon the idea that life is organized chemistry. But today, we take the next step together to explore how that organization actually looks. The cell is the bridge where the inanimate world of atoms and molecules crosses over into the vibrant, breathing world of biology. It is the smallest unit of life, a boundary between the chaos of the outside world and the exquisite order of the internal one. As we explore this together, try to picture the cell not as a flat diagram in a book, but as a three-dimensional, fluid, and incredibly busy masterpiece of engineering. To understand the cell, we must first understand its borders. Every cell, no matter its type, is defined by a plasma membrane. I want you to imagine this membrane not as a rigid, cold wall, but as a discerning, shimmering veil, a liquid mosaic that is constantly in motion. It is composed primarily of lipids, specifically phospholipids, which have a very peculiar nature. One end of the molecule loves water, and the other end fears it. This dual personality causes them to line up in a double layer, creating a barrier that is oily and flexible. This membrane is the cell's gatekeeper. It doesn't just block things out. It actively decides who is allowed to enter the sanctuary of the cell and who must stay behind in the extracellular world. It is through this thin, fatty layer that the cell communicates with its neighbors and senses the world around it. Inside this boundary lies the cytoplasm. If the membrane is the city wall, the cytoplasm is the bustling plaza where all the action happens. It is a crowded, organized soup made of a jelly-like substance called the cytosol, filled with proteins, salts, and the tiny machines we call organelles. Everything in the cytoplasm has a purpose and a place. It is not a random collection of parts. It is a highly choreographed dance of molecules moving through a structured framework. As we look closer at these cities of life, we notice that they come in two main designs. The first and most ancient, is the prokaryotic cell. These are the cells of bacteria and archaea. You might picture a prokaryotic cell as a simple, efficient studio apartment. There are no internal walls or separate rooms. The DNA, the master blueprint of the cell, just floats in a specific region called the nucleoid. Everything the cell needs to survive happens in that one open space. It is a design of incredible simplicity and resilience, one that has survived on Earth for billions of years. However, as life evolved, it sought more complexity, leading to the eukaryotic cell. These are the cells that make up your body, the trees outside your window, and the cat sleeping at the foot of your bed. If a prokaryote is a studio apartment, a eukaryote is a grand, sprawling mansion with many specialized rooms. These rooms are what we call organelles, or little organs. Each organelle is wrapped in its own membrane, allowing it to maintain its own unique environment. This compartmentalization is the secret to the eukaryote's success. It allows the cell to perform many different, sometimes conflicting chemical reactions all at once, without them interfering with one another. It is the ultimate expression of biological efficiency. Take a moment to let that idea find its place. The transition from a simple open space to a complex room-filled mansion. It is a journey of organization that mirrors the way we build our own civilizations. Now, let us walk through the rooms of this eukaryotic mansion and meet the residents. At the very center of the cell, we find the nucleus. This is the cell's library and command center. It is protected by its own double membrane, the nuclear envelope, and inside, it holds the most precious treasure of life, your DNA. This DNA isn't just a list of instructions, and it is an ancient manuscript, coiled and folded with immense care into structures called chromatin. When the cell needs to build something, it doesn't send the original DNA out into the busy cytoplasm. Instead, it makes a careful copy, a transcript called RNA, and sends that out to the workers. This ensures that the master blueprints are never lost or damaged. Nearby we find the mitochondria, often called the powerhouses of the cell. But I prefer to think of them as ancient golden lanterns. They are fascinating because they have their own DNA and a double membrane, which tells us a story of a deep, historical friendship. Billions of years ago, the ancestors of these mitochondria were likely independent bacteria that were swallowed by a larger cell. Instead of being digested, they stayed, providing the cell with energy in exchange for a safe place to live. This is the endosymbiotic theory, a reminder that life is built on cooperation as much as competition. The mitochondria take the energy from the food you eat and convert it into a molecular currency called ATP, which the rest of the cell can use to do work. Moving through the cytoplasm, we encounter a vast network of folded membranes, known as the endoplasmic reticulum, or the ER. The rough ER is studded with tiny hard-working structures called ribosomes. These ribosomes are the master builders. They read the RNA instructions from the nucleus and use them to assemble long chains of amino acids into proteins. These proteins are then sent into the smooth ER, where they are folded and modified, or where lipids are synthesized. Once these products are made, they need to be shipped to their final destinations. This is where the Golgi apparatus comes in. Think of it as the cell's post office. It receives packages from the ER, puts the finishing touches on them, and adds a molecular address label, before sending them off in tiny bubble-like transport vehicles called vesicles. And we cannot forget the cleaners. The lysosomes are the cell's recycling centers. They contain powerful enzymes that can break down waste, old organelles, and even invaders like bacteria. They ensure that the cell doesn't become cluttered with useless debris, turning old parts into new building blocks. This constant cycle of creation and destruction is the very heartbeat of metabolism. Finally, providing shape to the entire structure is the cytoskeleton. This is not a rigid skeleton of bone, but a dynamic, ever-changing web of protein fibers. It acts as both a scaffold for support and a system of tracks upon which the vesicles can travel, like tiny trains moving through the city. If your mind is still following along, try visualizing this: the library, the power plants, the factories, and the post office, all working in a quiet, rhythmic pulse of life. So you see, this isn't just a theory in a book. This cellular foundation is the reason medicine is possible. When you take an antibiotic, it often works by attacking the specific structures of a prokaryotic bacterial cell, like its unique cell wall, while leaving your own eukaryotic cells unharmed. It is a precise strike based entirely on the differences in cellular architecture. Furthermore, understanding the cell helps us understand the nature of disease. When we talk about cancer, we are talking about cells that have forgotten how to listen to the instructions in the nucleus, cells that begin to grow and divide without regard for the boundaries of the city. When we talk about aging, we are often talking about the gradual slowing of the mitochondria, or the accumulation of waste that the lysosomes can no longer clear. By studying the foundations of the cell, we are studying the foundations of our own health and longevity. This is the principle that explains why we are all connected. Every living thing you see, from the blade of grass to the person sitting next to you, shares this same fundamental logic. We are all built from the same types of rooms, the same types of blueprints, and the same types of energy. It is a beautiful, unifying truth of our existence. Having now grasped the structure of the cell, the grand mansion where life resides, we are perfectly prepared to appreciate the materials used to build it. We've looked at the city, but we haven't yet looked at the bricks and mortar in fine detail. We've mentioned lipids and proteins, but what are they made of? This is a good place to rest your thoughts for a moment before we continue. Let the image of the busy, glowing cell fade into the background of your mind. Our next lesson will hand us the adjacent piece of the puzzle, which is a study of the specific chemical elements that nature has chosen to weave into the fabric of life. We will look at the periodic table not as a chart of chemicals, but as a menu of possibilities that life uses to create the diversity we see around us. When you are ready, we will move from the architecture of the cell to the very atoms that hold it all together. Lesson 2: Chemical Elements and the Periodic Table of Life. In our first session, we wandered through the grand microscopic mansion of the cell, marveling at its rooms and the busy work being done within its walls. It was a journey of architecture and space. But now, we must look closer. We must zoom in past the organelles, past the membranes, until we reach the very substances that compose the walls and the workers themselves. In this lesson, we are going to explore the specific chemical elements that nature has curated for the masterpiece of life. Out of the more than 100 elements found in the periodic table, Life is remarkably selective, choosing only a small handful to perform the heavy lifting of existence. We often think of the periodic table as a daunting chart on a chemistry lab wall. But for our journey tonight, I want you to view it as a palette of colors. Just as a painter can create an infinite number of shades using only a few primary pigments, life uses a narrow selection of elements to build the staggering diversity of every organism that has ever walked, swum, or flown upon this earth. As we take this next step together, let the complexity of the world fall away, and let us find comfort in the elegant simplicity of the atoms that make us who we are. If we were to break down a human body or a towering oak tree or even a tiny blade of grass into its most basic ingredients, we would find that about 96% of that mass is made up of just 4 elements: hydrogen, oxygen, nitrogen, and carbon. We often call these the Big Four, or the bulk elements. They are the primary letters in the alphabet of biology. To understand why life is so fond of them, we must look at their individual personalities. Let us start with carbon, the undisputed protagonist of our story. If the cell is a mansion, carbon is the master architect and the primary building material all in one. Carbon is unique because of its 4 valence electrons, which is a fancy way of saying it has 4 hands reaching out, eager to hold on to other atoms. This allows carbon to form 4 strong, stable bonds at once. Because of this, carbon can build long, straight chains, or it can branch out into complex skeletons, or even wrap around itself to form sturdy rings. No other element can match carbon's versatility in building 3-dimensional shapes. Think of carbon as the ultimate Lego brick. Because it can connect in so many directions, it can create the vast, complex molecules like proteins and DNA that simpler elements just couldn't support. When we say we are carbon-based life forms, we are acknowledging that without the structural flexibility of the carbon atom, the intricate machinery of our bodies simply wouldn't have a frame to hang on. Next, we have hydrogen. Hydrogen is the simplest and most abundant element in the universe. In the context of life, you might think of hydrogen as the filler, or the constant companion. Because it only has 1 electron to share, it usually finds its place on the edges of carbon chains, capping them off and providing stability. But do not let its simplicity fool you. As we will see later in our journey, hydrogen plays a vital role in how we move energy and how we keep our internal environment balanced. It is the quiet, essential partner to every other element. Then there is oxygen. Oxygen is the breath of life, but in a chemical sense, it is the great attractor. It is highly electronegative, which means it has a very strong pull on electrons. When oxygen is part of a molecule, it often creates a tug-of-war that gives the molecule a specific electrical personality. This is why oxygen is so critical in the molecules that help us process energy from the food we eat. It is the final destination for electrons in our metabolic pathways, acting like a powerful magnet that keeps the flow of energy moving in the right direction. Finally, we have nitrogen. If carbon provides the skeleton and oxygen provides the energy flow, nitrogen provides the structural specificities of our most important tools, proteins and nucleic acids. Nitrogen is a key component of the amino acids that build our muscles and enzymes, and the nitrogenous bases that make up the rungs of our DNA ladder. It is an element that demands a certain level of respect, as it is often difficult for organisms to pull it out of the atmosphere and turn it into a form they can use. Yet, once it is inside the cell, it is indispensable for defining the functions that make life active and responsive. Take a moment to let that idea find its place. 4 simple elements: carbon, hydrogen, oxygen, and nitrogen, working in a constant, quiet partnership to build every muscle fiber, every nerve cell, and every strand of hair you possess. It is a profound economy of design. While the big 4 do most of the work, there are 2 other elements that we must mention to complete the set of primary biological building blocks: phosphorus and sulfur. Together with the others, they form the acronym CHNOPS. These 6 elements are the true periodic table of life. Phosphorus is the backbone of our energy currency, ATP, and the framework of our DNA. It is highly reactive and carries a strong negative charge, making it perfect for storing and releasing energy. Sulfur, on the other hand, is often found in the bridges that hold proteins in their specific shapes. Think of sulfur like a structural staple. It creates disulfide bonds that ensure a protein doesn't lose its shape when things get difficult. Now, if we step back and look at the periodic table as a whole, we might wonder why life didn't choose other, more common elements. For instance, a silicon sits right below carbon on the table and also has 4 hands for bonding. For decades, science fiction writers have imagined silicon-based life on other planets. However, silicon is heavier, and its bonds are not quite as flexible or as stable in water as carbon's bonds are. Carbon-carbon bonds are strong enough to hold a molecule together in a stormy environment, but gentle enough to be broken and rebuilt when the cell needs to change. Life, in its infinite wisdom, chose the element that provided the perfect balance between strength and adaptability. We should also pay our respects to the trace elements. These are elements that appear in tiny, almost invisible amounts within our bodies, yet life would cease to function without them. Consider iron. It makes up a tiny fraction of your body mass. Yet it is the very heart of the hemoglobin molecule in your red blood cells. It is the iron atom that catches the oxygen you breathe and carries it to your brain and your toes. Without that single atom of iron nestled in a bed of nitrogen and carbon, your blood would lose its ability to sustain you. Think also of iodine, which is used by your thyroid gland to regulate your metabolism, or zinc, which helps hundreds of different enzymes perform their chemical magic. We even have tiny amounts of copper, selenium, and magnesium. We call them trace because they are like the rare spices in a grand recipe. You only need a pinch, but without that pinch, the entire dish fails. These elements were discovered and categorized over centuries by scientists who realized that health often depended on things we couldn't even see. In the early 1900s, for example, researchers realized that a lack of simple iodine caused goiters, leading to the universal practice of adding iodine to table salt. This is a reminder that our connection to the Earth's crust is literal. We are composed of the very minerals that make up the stones in the soil, refined by billions of years of biological history. You might ask yourself why this matters as you drift toward sleep. It matters because this alphabet of life is the reason we can understand our place in the cosmos. When astronomers point their telescopes toward distant stars and planets, they aren't looking for little green men. They are looking for the chemical signatures of carbon, oxygen, and water. We look for these because we know that the chemical logic of these elements is universal. If life exists elsewhere, it likely follows the same foundational rules that you do. This knowledge also grounds us in our own environment. It explains why nutrition is so important. When we eat, we aren't just fueling up like a car. We are providing the raw materials for the constant renovation of our cellular mansion. Every few years, nearly every atom in your body is replaced by new atoms from the food you eat and the water you drink. You are, quite literally, a flowing river of elements. The carbon that was once in a piece of fruit becomes part of your muscle. The nitrogen in a bean becomes part of your DNA. We are in a constant state of exchange with the world around us. Don't worry about memorizing the atomic numbers or the exact weights of these elements. Just let the logic of it wash over you. The idea that the vast complexity of your life, your memories, and your dreams, your movements, is built upon a small, elegant set of pieces. It is a comforting thought, isn't it? To know that at your core, you are built from the most fundamental and beautiful materials the universe has to offer. We have explored the what of biochemistry, the elements that serve as our building blocks. We've seen how carbon acts as a frame, how oxygen draws in energy, and how trace elements add the finishing touches. But knowing what the bricks are made of is only half the story. The next question is, how do they stick together? How do these atoms reach out and grab one another to form the bonds that hold a molecule in place? Let's hold that thought right here, a complete piece of the puzzle. Our next lesson will hand us the adjacent piece, which is a study of the invisible forces and the handshakes between atoms. We will go deeper into the architecture of the atom itself to see how electrons create the glue of life. So, when we return, we will begin our study of the architecture of the atom and the nature of chemical bonding. For now, let your mind rest on the simplicity of the atoms. Feel the weight of your body supported by the earth, and know that you are a masterpiece of elemental design, perfectly assembled and quietly at peace. Take a deep breath, and as you exhale, imagine those tiny, ancient atoms within you, holding on with steady, silent strength. You are well made, you are secure, and you are ready for the next step in our journey. Lesson 3. The architecture of the atom and chemical bonding. In our last session, we spent some time admiring the alphabet of life, those specific elements like carbon, hydrogen, and oxygen that nature has chosen to build the grand mansion of the cell. We looked at them as individual entities like characters in a story waiting for the plot to begin. But in the world of biochemistry, no atom is an island. For life to exist, these atoms must interact, they must reach out, touch one another, and hold on with a strength that can withstand the chaotic environment of a living system. In this lesson, we are going to explore the invisible architecture that allows this to happen. We will go deep inside the atom to see how its internal structure dictates its external behavior, and we will look at the different types of handshakes, the chemical bonds that atoms use to stay connected. As we explore this together, I want you to imagine that we are looking at the invisible glue of the universe. It is a quiet, powerful force that turns a collection of dust into a breathing, thinking human being. Don't worry about the complexity of the physics. Instead, try to feel the logic and the balance that exists at the heart of every single molecule in your body. To understand how atoms stick together, we must first look at how they are built. Every atom is like a tiny solar system. At the very center, we have the nucleus, a dense core made of protons and neutrons. The protons carry a positive charge, while the neutrons are neutral, acting as a sort of buffer to keep the protons from pushing each other away. But for our story of biochemistry, the real magic happens on the outskirts of the atom, in the vast open space surrounding the nucleus. This is where the electrons live. I want you to picture the electrons not as little planets orbiting in perfect circles, but as a shimmering, energetic cloud. Electrons carry a negative charge, and they are constantly in motion, drawn toward the positive heart of the nucleus, but kept at a distance by their own incredible energy. In biochemistry, these electrons are the social butterflies of the atom. They are the only parts of the atom that ever actually touch the outside world. These electrons are organized into shells or layers, and each shell can only hold a certain number of guests. The first shell, closest to the nucleus, is small and can only hold 2 electrons. The second and third shells are larger and prefer to have 8. This leads us to a beautiful principle called the octet rule. Atoms, much like us, seem to crave a sense of wholeness or completion. An atom is at its most stable, its most peaceful, when its outermost shell is full. If an atom has 7 electrons in its outer shell, it feels an almost irresistible pull to find one more. If it has only one, it often feels the need to give that one away so it can drop down to a full shell underneath. This desire for a full outer shell is the driving force behind all chemical bonding. The most intimate way atoms find this completion is through the covalent bond. Imagine 2 atoms standing close to one another, both of them needing an extra electron to feel complete. Instead of fighting over one, they decide to share. They move so close that their electron clouds overlap, and the shared electrons begin to zip around both nuclei. This shared embrace is incredibly strong. It is the superglue of the biological world. Most of the molecules that make up your body, your proteins, your fats, your DNA, are held together by these covalent bonds. They are permanent, stable connections that don't easily break, which is exactly what you want when you are building something as complex as a person. But there is another way atoms can find stability, and that is through the ionic bond. Think of this not as a shared embrace, but as an attraction of opposites. Sometimes, one atom is so much stronger than another that it simply takes an electron for itself. This leaves the first atom with a positive charge, and the second atom with a negative charge. And because opposite charges attract, these 2 atoms, now called ions, stick together like powerful magnets. You see this most clearly in simple table salt, where sodium gives an electron to chlorine. They aren't sharing in the same way, but they are bound together by a profound electrical attraction. Take a moment to let that idea find its place. The 2 ways of being together. One is a deep, shared connection, and the other is a magnetic attraction. Both are essential, and both create the stability that life requires. If your mind is still following along, try visualizing those tiny clouds of energy overlapping, finding a point of perfect balance where they no longer need to search for anything more. As we add layers to this understanding, we encounter the work of a man named Linus Pauling, one of the greatest chemists of the 20th century. He introduced us to the concept of electronegativity. This is a simple but powerful idea. It is a measure of how greedy an atom is for electrons. Not all atoms share equally. If you have a covalent bond between two identical atoms, like two carbons, they share the electrons perfectly in the middle. We call this a nonpolar covalent bond. However, if one atom is much more electronegative than the other, if it is much greedier, it will pull the shared electrons closer to its own side. Think of it like a game of tug-of-war, where one person is much stronger than the other. The rope stays connected, but most of it is on the stronger person's side. This creates a polar covalent bond. The greedy atom becomes slightly negative because the electrons are spending more time there, and the other atom becomes slightly positive. This tiny difference in charge is one of the most important concepts in all of biochemistry. It is the reason water behaves the way it does, and it is the reason your proteins can fold into complex shapes. Oxygen is one of the greediest atoms in the biological world, while hydrogen is one of the most generous. When they bond together, they create a polar molecule that is always looking for a way to interact with its neighbors. We should also consider the bond energy. Breaking a covalent bond requires a significant amount of energy, like trying to pull apart two pieces of wood that have been bolted together. On the other hand, ionic bonds in a watery environment, like inside your cells, are much easier to manipulate. This difference in bond strength allows life to be both sturdy and flexible. Your DNA is held together by strong covalent bonds along its length, so the information is never lost. But the two strands are held to each other by much weaker forces that can be unzipped when it's time to read the instructions. It is a masterpiece of molecular engineering. Don't worry about memorizing the specific electronegativity values or the kilojoules of energy involved. Just let the logic of it wash over you. Life uses a variety of fasteners, some as strong as bolts, some as flexible as Velcro, to ensure that every part of the cell can do exactly what it needs to do at exactly the right time. This isn't just a theory in a book. It is the principle that explains why you don't dissolve when you step into a bathtub. The covalent bonds that hold your skin cells together are far too strong to be bothered by the water. However, the ionic bonds in the salt you might add to that bath dissolve almost instantly. The water molecules, with their polar tug-of-war personalities, swarm around the salt ions and pull them apart. This also explains the stability of the food you eat. When you eat a piece of bread, your body has to use specialized tools, enzymes, to break those strong covalent bonds in the starch and turn them into sugar that your cells can use. This process releases the energy that was stored in those bonds. In a very real sense, the energy you use to walk, talk, and think is the energy that was once holding atoms together in a plant or an animal. You are powered by the breaking of chemical bonds. So you see, this architecture of the atom is the foundation of everything you experience. It is why medicines can bind to specific receptors in your brain to take away a headache. And it is why the air you breathe stays inside the hemoglobin of your blood until it reaches the cells that need it. Every breath, every thought, every heartbeat is a celebration of these invisible handshakes. We have looked at the how of molecular connection, the bonds that act as the glue of life. We've seen how atoms seek wholeness through the sharing or transferring of electrons, and how the greediness of certain atoms creates a world of electrical polarities. But there is one more dimension we must consider. Atoms don't just bond in a flat, two-dimensional world. They exist in 3 dimensions. The way they are arranged in space, the angles of their bonds, and the shapes they form is just as important as the bonds themselves. In our next lesson, we are going to explore the 3-dimensional dance of molecules. We will look at molecular geometry and the fascinating world of isomerism, where 2 molecules can have the exact same atoms and the exact same bonds, but because they are shaped differently, they behave in completely different ways. This is a good place to rest your thoughts for a moment before we continue. Let the image of those overlapping electron clouds stay with you as you rest. Our next lesson will take these building blocks and show you how they are assembled into the specific, intricate shapes that allow life to recognize itself. So, when we return, we'll begin our study of the shapes of life. For now, let yourself feel the stability of the atoms that make you. You are held together by forces that are ancient, reliable, and perfectly balanced. Take a deep, slow breath, and as you drift into a deeper state of relaxation, know that the very architecture of your being is working in perfect harmony even while you sleep. We will be here, ready to take the next step together whenever you are. Lesson 4. Molecular Geometry and Isomerism. In our last session, we spent some time admiring the invisible handshakes that hold the world together, the covalent and ionic bonds that act as the glue for our elemental building blocks. We understood how atoms seek a sense of wholeness by sharing or trading their electrons. But if we stop there, we are only looking at a flat, two-dimensional map of a three-dimensional world. In this lesson, we are going to step into the 3rd dimension. We are going to explore molecular geometry and the fascinating world of isomerism. Think of this as the transition from looking at a blueprint to walking through the actual house. In biochemistry, the identity of a molecule is not just defined by what atoms it contains, but by exactly where those atoms are sitting in space. A single shift in position, a tiny rotation of a bond, or a mirror image reflection can change a life-saving medicine into a silent bystander or even a poison. As we explore this together, I want you to visualize these molecules not as static letters on a page, but as elegant, tumbling dancers in a vast, microscopic ballroom. The shape of the dance is what gives life its nuance and its specificity. To understand why molecules take the shapes they do, we have to remember the personality of the electron. Electrons, as we've discussed, are negatively charged clouds of energy. And because like charges repel each other, these electron clouds want to stay as far apart as possible. This is the heart of what scientists call valence shell electron pair repulsion, or VSEPR theory. You don't need to remember the name, but I want you to remember the logic. Imagine you are holding a cluster of balloons tied together at the strings. If you try to push 2 balloons together, the others will naturally shift and push away to create space. Atoms do the exact same thing. When a carbon atom forms 4 bonds, those 4 bonds push away from each other until they are as far apart as they can possibly be. This creates a shape called a tetrahedron. In a tetrahedron, the central carbon sits in the middle, and the 4 atoms it is bonded to sit at the corners of a triangular pyramid. If you were to measure the angle between these bonds, you would find it is exactly 109.5 degrees. This isn't just a random number. It is the mathematical sweet spot of maximum distance. This three-dimensional tetrahedral shape is the foundation of almost all organic chemistry. It allows carbon to build complex, sturdy structures that aren't just flat ribbons, but intricate, jagged, and beautiful sculptures. Once we understand that molecules have specific shapes, we encounter a strange and wonderful phenomenon called isomerism. The word comes from the Greek isos, meaning equal, and meros, meaning part. Isomers are molecules that have the exact same parts, the same number of carbons, hydrogens, and oxygens, but they are put together in a different order or a different orientation. I want you to think of a set of children's building blocks. If you have 10 red blocks and 5 blue blocks, you could build a tall, skinny tower, or you could build a short, wide bridge. The ingredients are identical, but the result is entirely different. In the world of molecules, we have structural isomers, where the atoms are literally plugged into different spots. One arrangement might be a straight chain, while another is a branched tree. Even though they have the same formula, they have different boiling points, different smells, and different ways of reacting with your cells. But there is a deeper, more subtle level of isomerism, called stereoisomerism. This is where the atoms are connected in the exact same order, but their orientation in space is different. This brings us to one of the most beautiful concepts in science, chirality, or handedness. I want you to take a moment and look at your own hands. Place them palms up in front of you. They look identical, don't they? They have the same number of fingers, the same structure, the same skin. But now, try to slide your left hand directly on top of your right hand, on both palms facing down. You'll notice they don't match. Your left thumb is where your right pinky should be. Your hands are mirror images of each other, but they are non-superimposable. You cannot turn your left hand into your right hand without essentially taking it apart and putting it back together backwards. In biochemistry, we call these mirror image molecules enantiomers. This happens whenever a carbon atom is bonded to 4 different things. That carbon becomes a chiral center, a point of asymmetry. In the mirror, the arrangement of those 4 groups is reversed. And while they might look the same to a casual observer, to the machinery of your body, the difference is profound. If your mind is still following along, try visualizing a molecule spinning in front of a mirror, and then imagine trying to pull that reflection out of the glass and fit it perfectly into the original. It simply won't fit. This is the subtle geometric language that life uses to distinguish one molecule from another. The history of chirality is one of the great detective stories of science, and it begins with a young Louis Pasteur in 1848. Before he became famous for pasteurization and vaccines, he was a chemist studying crystals that formed in wine barrels. He noticed that certain crystals of tartaric acid were shaped like little asymmetrical prisms. To his amazement, he saw that there were 2 types of crystals, some that tilted to the right and others that tilted to the left. With a pair of tweezers and a magnifying glass, he painstakingly sorted them into 2 piles. He discovered that when he dissolved these crystals in water, one solution would rotate a beam of polarized light to the right, and the other would rotate it to the left. He realized that the molecules themselves must be handed. This was the birth of stereochemistry. He famously remarked that life is dominated by asymmetrical actions, and he was right. We now know that almost all of the fundamental molecules of life are homochiral, meaning they only exist in one hand, in nature. For reasons we still don't fully understand, all the amino acids that build your proteins are left-handed, L-isomers, while almost all the sugars that provide your energy are right-handed, D-isomers. Why does this matter? It matters because of the lock and key mechanism. Your body is filled with receptors, proteins that act like locks. These locks are themselves asymmetrical and chiral. Imagine your right hand is a molecule trying to fit into a glove. A right-handed glove, the receptor, fits perfectly. But if you try to put your left hand into that same glove, it won't work. The wrong enantiomer cannot trigger the biological response because its shape doesn't match the three-dimensional negative space of the receptor. This is how your body ensures that only the right messages are sent and received. There is also a category of stereoisomers called diastereomers. These are molecules that have more than one chiral center. They aren't mirror images of each other. They are just different in some places and the same in others. Think of it like a pair of shoes where the left shoe is a size 8 and the right shoe is a size 10. They are related, but the symmetry is broken in a more complex way. These subtle differences in geometry are what allow life to create an almost infinite variety of structures using the same basic chemical toolkit. The importance of molecular geometry and chirality is perhaps most visible, and most tragic, in the world of medicine. You may have heard of the drug thalidomide. In the late 1950s, it was prescribed to pregnant women to treat morning sickness. What the scientists at the time didn't fully realize was that the drug was a mixture of two enantiomers, a right-handed version and a left-handed version. One version was a safe and effective sedative. However, its mirror image, the other hand, was a powerful teratogen, meaning it interfered with the development of the fetus. Because the body can sometimes convert one hand into the other, the results were devastating. This tragedy changed the way we test and regulate drugs forever. Today, pharmaceutical companies often go to great lengths to produce single enantiomer drugs. For example, if you take the common pain reliever, ibuprofen, you are taking a mixture of two hands, but only one of them actually works to stop your pain. The other hand is harmless, but it doesn't do anything. It's just along for the ride. In other cases, like the medicine albuterol used for asthma, using only the correct hand can reduce side effects and make the drug more effective. You can even experience chirality in your own kitchen. Consider the molecules limonene and carvone. One enantiomer of limonene smells like bright, fresh oranges, while its mirror image smells like lemons. An even more striking example is carvone. One hand of carvone gives spearmint its cool, refreshing scent. The mirror image of that exact same molecule is what gives caraway seeds their pungent, earthy smell. Your nose is actually a sophisticated chiral detector. It has different locks that recognize the right-hand mint and the left-hand caraway. So you see, geometry isn't just a subject for a math class. It is the very essence of how you perceive the world. It is the difference between the scent of an orange and a lemon, the difference between a medicine and a toxin, and the reason your body can read the instructions of your DNA with such perfect accuracy. Take a moment to let that idea find its place. The world as a collection of mirror images and three-dimensional puzzles. We have moved from the atoms themselves to the shapes they form in space. We've seen how the simple repulsion of electrons creates a tetrahedral world, and how the handedness of molecules defines the specificity of life. But a skeleton, even a three-dimensional one, needs some decoration to truly become functional. A carbon chain is like a blank canvas. To make it do something, we need to add specific tools or attachments to that frame. In our next lesson, we are going to explore functional groups. These are the specific clusters of atoms that we plug into our carbon skeletons to give them their chemical personality. They are the reasons why some molecules are acidic, why some are sweet, and why some can carry energy. Let's hold that thought here, a complete piece of the puzzle. Our next lesson will hand us the adjacent piece, which is the study of the tools of biochemistry. This is a good place to rest your thoughts for a moment before we continue. If you are still awake, try visualizing your hands again and imagine the thousands of tiny mirror image molecules inside you, finding their perfect locks and keeping the dance of life in motion. Don't worry about the details. Just let the logic of it wash over you. You are a masterpiece of geometry, a three-dimensional wonder held together by the perfect balance of space and form. Sleep well, knowing that every part of you is in its right and proper place. When you wake, or when we continue, we will look at how those shapes are brought to life by the functional groups that define their destiny. Lesson 5. Functional groups in biological molecules. In our journey through the microscopic world, we have spent much of our time admiring the architecture of life, the sturdy, versatile carbon skeletons, and the elegant three-dimensional geometry that gives molecules their shape. We've looked at the frame of the house, so to speak, but a frame, no matter how perfectly constructed, is not yet a home. It lacks the utility of a kitchen, the warmth of a fireplace, or the function of a doorway. In this lesson, we are going to explore the tools and decorations that turn a simple carbon chain into a functional, reactive participant in the dance of life. These are what we call functional groups. Think of a functional group as a specific cluster of atoms that, when plugged into a carbon skeleton, gives that molecule a distinct chemical personality. These groups are the reason some molecules are sweet, why others are sour, why some dissolve in water while others repel it, and why some can carry the explosive energy needed to move your muscles. As we explore these groups together, try to picture them as the various attachments on a Swiss Army knife. The carbon skeleton is the handle, the reliable steady base, while the functional groups are the blades, the scissors, and the files that actually perform the work. We are moving now from the study of what is to the study of what does. To understand functional groups, we must first realize that most carbon-to-carbon and carbon-to-hydrogen bonds are quite stable and, frankly, a bit boring. They don't like to react. They are content just to exist as a steady framework. However, when we replace one of those hydrogens with a functional group, everything changes. The first group we should meet is the hydroxyl group, composed of an oxygen atom bonded to a hydrogen atom, often written as -OH. When you add a hydroxyl group to a carbon chain, you create an alcohol. You might picture the hydroxyl group as a tiny polar magnet. Because oxygen is much greedier for electrons than hydrogen, it pulls the electrical charge toward itself, creating a small region of negativity. This makes the molecule water-loving or hydrophilic. It is the reason that sugars, which are covered in hydroxyl groups, dissolve so easily in your morning tea. Without these simple oxygen-hydrogen pairs, the complex carbohydrates that fuel your brain would be nothing more than inert, oily chains. Next, let us consider the carbonyl group, and where an oxygen atom is double bonded to a carbon atom. This group is a master of reactivity. If the carbonyl is at the end of a carbon chain, we call the molecule an aldehyde. If it's tucked into the middle, it's a ketone. This double bond to oxygen creates a point of intense chemical tension. It is like a spring waiting to be released. In the world of biochemistry, carbonyl groups are the key to the way our bodies break down sugars for energy. They are the reactive sites where bonds are broken and new ones are formed, allowing the cell to harvest the energy stored within. Take a moment to let that idea find its place. The transition from a passive chain to a reactive tool. If your mind is still following along, try to visualize the carboxyl group. And this is a fascinating combination. A carbon atom double bonded to an oxygen, like a carbonyl, but also single bonded to a hydroxyl group. We write this as -COOH. The carboxyl group is the acid of the biological world. It has a remarkable ability to let go of its hydrogen ion, releasing a positive charge into the surrounding environment. This is why we call them organic acids. You find this group in every single amino acid, the building blocks of your proteins, and in the fatty acids that make up your cell membranes. It is a group defined by its willingness to give, a characteristic that allows it to form strong ionic bonds and participate in the complex chemistry of protein folding. On the opposite side of the spectrum, we find the amino group, written as NH2. While the carboxyl group is an acid that gives away hydrogen ions, the amino group acts as a base, reaching out to pick them up. It consists of a nitrogen atom, bonded to two hydrogens. This group is essential for the structure of proteins. In every amino acid, you have a carboxyl acid at one end and an amino base at the other. This creates a beautiful, natural symmetry, an odd couple, that allows amino acids to link together in long, sturdy chains. The amino group is also what gives certain biological molecules their fishy or pungent scent, a reminder that chemistry is something we experience with all our senses. We also have the sulfhydryl group, which is a sulfur atom bonded to a hydrogen. While it might seem simple, this group is the staple of the protein world. When two sulfhydryl groups find each other in a long protein chain, they can form a disulfide bridge, a strong covalent bond that locks the protein into a specific shape. Think of it like the cross links in a fence. This is what gives your hair its strength and its texture. In fact, when someone gets a permanent at a hair salon, they are using chemicals to break and reform these sulfhydryl bonds to change the shape of their hair. Perhaps the most energetic of all is the phosphate group. Imagine a phosphorus atom surrounded by 4 oxygen atoms, carrying a heavy negative charge. This group is bulky, aggressive, and highly reactive. Because it is so negatively charged, putting 2 or 3 phosphate groups next to each other is like trying to push The same poles of two magnets together, they want to fly apart. This repulsion is exactly what makes ATP, the energy currency of your cells, so powerful. And when the cell clips off one of these phosphate groups, it releases a burst of energy that can power everything from the blink of an eye to a sudden sprint. It is the high-voltage battery of life. Finally, we have the methyl group, a carbon bonded to 3 hydrogens. Unlike the others, the methyl group is nonpolar and very stable. It doesn't like to react with water. Instead, the methyl group acts as a tag or a silencer. In the fascinating world of epigenetics, your cells add methyl groups to your DNA to turn certain genes off. It is like a molecular do not disturb sign, telling the cell's machinery to skip over that particular set of instructions. It is a reminder that in biochemistry, silence and stability are just as important as reactivity and energy. As we delve deeper into the significance of these functional groups, we must appreciate the work of the chemists and biologists who first decoded this molecular language. In the late 19th and early 20th centuries, researchers began to realize that the life force people had talked about for millennia was actually a series of very specific, very logical chemical interactions. They discovered that by changing just one functional group, they could completely alter the effect of a molecule on the human body. Consider the difference between the hormones testosterone and estrogen. At their core, these two molecules have almost identical carbon skeletons, 4 interlocking rings of carbon. However, the difference between them lies in the functional groups attached to those rings. Estrogen has a hydroxyl group where testosterone has a carbonyl group, and testosterone has an extra methyl group that estrogen lacks. And these tiny microscopic differences, just a few atoms shifted or changed, are responsible for the vast array of biological differences between the sexes. It is a profound example of how nature uses a very small alphabet of functional groups to write incredibly different stories. Image comparing the molecular structures of testosterone and estrogen, highlighting functional groups. We must also talk about the concept of solubility. Life, as we know it, is an aqueous phenomenon. It happens in water. But not every molecule wants to be in water. This creates the hydrophobic and hydrophilic divide. Functional groups like hydroxyl, carboxyl, and amino are the socialites. They love water and interact with it constantly. Methyl groups and long carbon chains are the hermits. They shy away from water. This tension between water-loving and water-fearing groups is what forces large molecules like proteins and cell membranes to fold and organize themselves. If your mind is still following along, try visualizing a long protein chain. It has different functional groups sticking out like various charms on a bracelet. As this chain moves through the watery environment of the cell, the hermit groups hide in the center to stay dry, while the socialite groups stay on the outside to touch the water. This seemingly simple game of hide and seek is what gives life its 3-dimensional complexity. This is the why behind the how. The functional groups aren't just there for decoration. They are the drivers of biological organization. So you see, this isn't just a theory in a textbook. It's the principle that explains the world you experience every day. When you smell the sharp, clean scent of a lemon, you are detecting the hydroxyl and carbonyl groups of molecules like citric acid. When you taste the sourness of vinegar, your tongue is reacting to the hydrogen ions being released by the carboxyl groups in acetic acid. Even the kick you get from your morning coffee is due to the way the functional groups on the caffeine molecule fit into the locks of the receptors in your brain. In the world of modern medicine, this knowledge is our greatest tool. Pharmacologists are essentially molecular decorators. If they have a drug that works well but doesn't dissolve in the blood easily, they might add a hydroxyl group to make it more water soluble. If a drug is broken down too quickly by the liver, they might mask a reactive site with a methyl group to make it last longer. Every medicine in your cabinet is a testament to our understanding of how these functional groups behave. This is also why nutrition is so vital. When you eat proteins, your body breaks them down into amino acids, harvesting those precious amino and carboxyl groups so it can rebuild your own unique tissues. You are, in a very literal sense, a collection of functional groups in constant motion. You are taking the tools from your environment and using them to maintain the mansion of your body. Take a moment to let that sink in. Don't worry about memorizing every structure or every name. Just let the logic of it wash over you. Life is a masterpiece of modular design. By using a few simple attachments on a carbon frame, nature has created the infinitely complex, beautiful, and responsive system that is you. We have explored the tools of biochemistry, the functional groups that provide the reactivity, the energy, and the personality to our molecular world. We've seen how they interact, how they define solubility, and how they allow life to be organized and purposeful. But there is one final piece of the environmental puzzle we must address. All of these functional groups, all of these bonds, and all of these geometries exist within a very specific medium. Life does not happen in a vacuum, nor does it happen in a solid block of stone. It happens in the most extraordinary substance in the universe, a substance that we often take for granted because it is everywhere. I am talking, of course, about water. Water is not just a passive background for these reactions. It is an active participant. Its unique physical and chemical properties are the reason these functional groups behave the way they do. Let's hold that thought here, a complete piece of the puzzle. Our next lesson will hand us the adjacent piece, which is a deep dive into the physical properties of water and why it is the miracle solvent that makes life possible. This is a good place to rest your thoughts for a moment before we continue. If you are still awake, try visualizing the molecules inside you, each one with its own set of tools, its own personality, and its own purpose, all floating in the gentle, supportive sea of your internal environment. You are a wonder of chemical coordination. Every functional group is in its place doing its work so that you can rest, sleep well, and when we return, we will plunge into the watery world that brings all of this chemistry to life. Lesson 6. The physical properties of water. We have spent our time thus far discussing the building blocks, the atoms, the bonds, and the geometric shapes that provide the structural logic of life. But imagine for a moment that we had all these perfectly formed pieces, the carbon skeletons, the amino groups, the phosphate batteries, and we simply laid them out on a dry, barren table. They would sit there, inert and silent. For the dance of life to actually begin, these molecules need a medium. They need a stage that is not just a passive floor, but an active participant that coaxes them into action. That medium, the very matrix of our existence, is water. In this lesson, we are going to explore the physical properties of H2O. It is a substance so common that we often forget it is a chemical miracle. And it is the only substance on Earth that exists naturally in all three states: solid, liquid, and gas, within the narrow range of temperatures that support life. As we explore the stickiness, the thermal resilience, and the strange density of water together, I want you to feel a sense of gratitude for this simple molecule. It is the gentle, supportive sea in which every one of your cells is currently bathed. Let us look at why water is not just a liquid, but the essential cradle of biochemistry. To understand why water is so special, we must return to the shape of the molecule itself. You know it as H2O, one oxygen atom bonded to two hydrogen atoms. Because of the tetrahedral geometry we discussed in our earlier lessons, you might expect it to be a straight line, but it isn't. And the oxygen atom has two lone pairs of electrons that act like invisible bulky clouds, pushing the two hydrogen atoms together. This gives water a distinct V shape with a bond angle of approximately 104.5 degrees. This shape is the first key to its magic. Now, remember our discussion about electronegativity, the greediness of atoms for electrons. Oxygen is one of the greediest atoms in the universe. It pulls the shared electrons away from the small, generous hydrogen atoms. Because the electrons, which carry a negative charge, spend more time near the oxygen, that end of the molecule develops a partial negative charge. Conversely, the hydrogen ends are left with a partial positive charge. This creates what we call a dipole. Water is a polar molecule. It has a north pole and a south pole of charge. Because opposites attract, the positive hydrogen end of one water molecule is drawn to the negative oxygen end of another. They reach out and perform a gentle, fleeting handshake called a hydrogen bond. I want you to picture this not as a permanent weld, but as a momentary embrace. In liquid water, these bonds are constantly breaking and reforming, lasting only a trillionth of a second. But because there are so many of them, they create a collective stickiness that we call cohesion. This is why water forms droplets on a window pane rather than spreading out into a thin, dry film. It is also the reason for surface tension. At the surface of a lake, the water molecules have no neighbors above them to hold on to, so they pull more tightly to the ones beside and below them. This creates a sort of invisible skin strong enough to support the weight of a water strider or a fallen leaf. But water doesn't just stick to itself. It also sticks to other things, a property we call adhesion. If you've ever seen water climb up a thin glass tube or watched a paper towel soak up a spill, you are seeing adhesion and cohesion working together in a process called capillary action. Within you, this is how water moves through the tiniest vessels of your body. And in the world outside, it is how a 300-foot redwood tree pulls moisture from the soil all the way to its highest leaves against the relentless pull of gravity. Take a moment to let that logic find its place. The V shape leading to the dipole leading to the handshake, and the handshake leading to the strength of a forest. If your mind is still following along, try to visualize those trillions of molecules within you, constantly reaching out, letting go, and reaching out again in a rhythmic, liquid pulse. As we go deeper, we encounter the thermal properties of water, which act as the great stabilizer of our planet and our bodies. Water has an incredibly high specific heat. In simple terms, this means it takes a vast amount of energy to raise the temperature of water even by 1 degree. Think of a metal pan on a stove. Within seconds of turning on the heat, the metal is too hot to touch. But the water inside the pan stays cool for several minutes. This is because most of the heat energy is being used up just to break those handshakes, the hydrogen bonds, before the molecules can start moving faster and raising the temperature. For you, this is a life-saving property. Your body is about 60 to 70% water. Because water resists temperature changes, you can walk from a cold room into a warm one without your internal chemistry going into a shock. It keeps your core temperature stable, allowing the delicate enzymes we will study later to function without interruption. On a global scale, the oceans act as a massive heat sink, absorbing the sun's energy during the day and releasing it slowly at night, preventing the Earth from swinging between the extremes of boiling and freezing. Related to this is the heat of vaporization. To turn liquid water into steam, you have to break every single hydrogen bond. This requires an enormous amount of energy. When you sweat, your body uses this principle to cool you down. As the water evaporates from your skin, it carries a massive amount of stolen body heat away with it into the air. It is the most efficient cooling system ever designed, and it is entirely dependent on the strength of those tiny molecular handshakes. Perhaps the most poetic and strange property of water is what happens when it freezes. Most substances get denser as they get cold. The molecules slow down and huddle closer together until they sink. If water behaved like most other liquids, the oceans would freeze from the bottom up, eventually becoming solid blocks of ice that would never melt, and life would have nowhere to hide. But water is different. As water cools toward 4 degrees Celsius, it behaves normally, getting denser. But as it drops below 4 degrees Celsius and heads toward freezing, something miraculous happens. The molecules slow down enough that the hydrogen bonds become permanent. Because of that 104.5 degrees angle we mentioned, the molecules are forced to lock into a wide, airy, hexagonal lattice. They actually push each other further apart to make room for the bonds. This makes ice less dense than liquid water. This is why ice floats. It creates an insulating blanket on the top of a lake, keeping the liquid water below at a steady, life-sustaining temperature even in the harshest winter. It is a fundamental law of the universe that seems specifically designed to protect the living. The final property we must discuss is water's role as the universal solvent. Because water is polar, it is incredibly good at dissolving other polar substances and ionic compounds like salt. When you drop a crystal of salt into water, the negative oxygen ends of the water molecules swarm around the positive sodium ions, and the positive hydrogen ends swarm around the negative chloride ions. They surround the ions in what we call a hydration shell, gently pulling them away from the crystal and into the solution. This ability to dissolve and transport substances is why your blood is water-based. It allows your body to carry oxygen, nutrients, hormones, and waste products to exactly where they need to go. If your internal environment were made of oil or alcohol, the sheer variety of life's chemistry would come to a grinding halt. Water is the only solvent that can accommodate the incredible diversity of biological molecules. Think of this the next time you take a sip of water or feel a raindrop on your skin. You aren't just looking at a simple liquid. You are looking at a substance whose every physical property, its stickiness, its refusal to heat up quickly, its ability to float when it's cold, and its capacity to dissolve the world is perfectly tuned to the requirements of life. This isn't just theory. It is the physical reality that allows you to exist. Don't worry about memorizing the bond angles or the specific heat values. Just let the beauty of the design wash over you. You are a creature of water, sustained by a molecule that is as humble as it is extraordinary. Every chemical reaction we discuss from here on out is only possible because it is happening in this gentle, supportive, and active sea. We have explored the stage upon which life performs, the miracle of water and its unique physical properties. We've seen how the dipole and the hydrogen bond create a world of stability and transport. But as we move forward, we must look at how other molecules interact with this watery stage. Not every molecule is a friend of water, and not every interaction is a permanent bond. In our next lesson, we are going to explore the world of non-covalent interactions in aqueous systems. We've seen the handshakes of water, but what about the hugs and shoves between other molecules? We will look at how the presence of water forces proteins to fold, and how it creates the boundaries of our cells through the hydrophobic effect. Let's hold that thought here, a complete piece of the puzzle. Our next lesson will hand us the adjacent piece, which is the study of the invisible forces that govern the crowded, watery interior of the cell. This is a good place to rest your thoughts for a moment before we continue. If you are still awake, try to visualize the water within you, calm, steady, and holding everything in its proper place. You are a wonder of hydration and balance. As you rest, let the quiet strength of these molecular handshakes give you a sense of security. Your internal sea is at peace, and you are exactly where you need to be. When we return, we will dive deeper into the forces that shape the very molecules of life within that sea. Sleep well, and I will be here when you are ready to continue. Lesson 7. Non-covalent interactions in aqueous systems. In our previous session, we spent some time reflecting on the miracle of water, that simple polar molecule that provides the stage for everything we are. We looked at its stickiness, its thermal resilience, and its role as the universal solvent. But now, we must look at who else is on that stage and how they interact with the water and with each other. If the covalent bonds we discussed earlier are the superglue of life, strong, permanent, and difficult to break, then the interactions we are exploring today are the Velcro. These are the non-covalent interactions. They are individually weak, often fleeting, and easily disrupted by a change in temperature or a shift in the environment. Yet, do not let their individual weakness deceive you. In the crowded, bustling interior of a cell, these weak forces are the most powerful architects of all. They are the reason your DNA can unzip to be read and then zip back up with perfect precision. They are the reason a protein, which starts as a simple string of amino acids, can fold itself into a complex, three-dimensional machine. As we walk through this together, I want you to imagine a crowded room where everyone is gently bumping into one another, sometimes shaking hands, sometimes offering a brief hug, and sometimes politely moving out of the way. This subtle, constant social dance of molecules is what we call non-covalent interaction. It is the soft logic that allows life to be dynamic, responsive, and infinitely complex. To understand this molecular dance, we must first distinguish between the four main types of handshakes that occur in the watery world of the cell. The first, which we have already touched upon, is the hydrogen bond. While we saw how water molecules bond to each other, in this lesson, we see how they bond to other things. Any molecule that has a greedy oxygen or nitrogen atom attached to a generous hydrogen atom can form a hydrogen bond with water or with another molecule. Think of these as the most common greetings in the cell. They are like a gentle touch on the shoulder, enough to hold two things together for a moment, but easy to let go of when the situation changes. The second type of interaction is the ionic interaction, sometimes called a salt bridge or a Coulombic interaction. This happens between two atoms that have full opposite charges. Imagine a positively charged amino group and a negatively charged carboxyl group. Because opposites attract, they pull toward each other with a force that is stronger than a hydrogen bond, but still not as permanent as a covalent bond. These are the firm handshakes of the cellular world. They provide structural stability to proteins, acting like invisible anchors that keep distant parts of a long molecule tucked neatly together. Then, we have the most subtle of all, the van der Waals interactions. These are the fleeting glances of biochemistry. They occur between all atoms, even those that have no charge and no polarity. Because electrons are always moving in a random shimmering cloud for a tiny fraction of a second, an atom might have more electrons on one side than the other. This creates a transient dipole, a temporary magnet. For that split second, it can attract a neighboring atom. These forces are incredibly weak, but when thousands of atoms are packed closely together, like the layers of a blooming flower, the sum of these tiny glances becomes a significant force that helps hold the structure together. Finally, we must discuss the most important driver of biological structure, the hydrophobic effect. This is not so much a bond as it is a shove. I want you to picture a crowded elevator filled with people who all know each other. These are the water molecules, busy forming their hydrogen bond handshakes. Now, imagine a stranger walks in who doesn't speak the language and refuses to shake hands. This is a non-polar, water-fearing molecule, like a droplet of oil. The water molecules don't want to interact with the stranger, but they also don't want to lose their own connections. To keep their handshakes going, the water molecules are forced to form a highly organized, cage-like structure around the stranger to keep it separate. This cage is very restrictive and requires a lot of order, which, as we learned in our lesson on entropy, the universe dislikes. To regain their freedom, the water molecules shove all the strangers together into one group. By huddling the oil droplets together, the water molecules minimize the surface area they have to cage, allowing more water molecules to move freely. This shoving of non-polar molecules into the center of a structure, away from the water, is the single most powerful force that causes cell membranes to form and proteins to fold into their functional shapes. Take a moment to let that idea find its place. The shove of the water molecules, forcing order by seeking their own freedom. If your mind is still following along, try to visualize the interior of your cells not as a static soup, but as a sea of millions of these interactions happening every microsecond. It is a world of constant movement, where the Velcro is being pressed together and pulled apart in a rhythmic, life-sustaining pulse. To truly appreciate the significance of these weak interactions, we must look at how they work in concert. A single hydrogen bond is like a single thread of silk. You could break it with the slightest effort. But when you weave thousands of those threads together, you create a fabric strong enough to stop a falling weight. This is the secret of cumulative strength. Consider a protein. A protein is often a long, floppy string of hundreds of amino acids, and to do its job, whether that's carrying oxygen or breaking down sugar, it must fold into a very specific, precise, three-dimensional shape. If it were held together by strong covalent bonds, it would be a rigid, unmoving statue. But instead, it is held together by thousands of these non-covalent interactions. The water-fearing parts are shoved into the dark, dry center by the hydrophobic effect. The water-loving parts stay on the outside, forming hydrogen bonds with the surrounding cellular sea. Along the way, ionic anchors and van der Waals glances lock the loops and coils into place. This creates a structure that is stable, yet flexible. It can breathe. It can change shape slightly when a signal arrives, then snap back into place. This conformational change is the basis of all biological signaling and movement. Now, as if our molecules were held together by superglue, we would be as inert as stones. It is the Velcro that allows us to be alive. Historically, it took scientists a long time to realize that weak could mean important. In the early days of chemistry, the focus was entirely on the strong bonds that define a molecule's identity. It wasn't until researchers like Linus Pauling and Max Perutz began to solve the structures of complex proteins like hemoglobin that the true power of non-covalent interactions became clear. They realized that the magic of life wasn't in the atoms themselves, but in the subtle, non-permanent way those atoms arrange themselves in space. We must also consider the concept of complementarity. For two molecules to stick together using these weak forces, they must fit together like two pieces of a jigsaw puzzle. Their shapes must match so that the atoms can get close enough for van der Waals forces to act, and their charges must be lined up so that positive meets negative. This molecular recognition is how your immune system knows the difference between a healthy cell and a virus. It is how an enzyme finds its specific key in a sea of millions of other molecules. It is a world of incredible precision, built entirely on the most delicate of forces. You might wonder how this microscopic shoving and handshaking affects the world you see. One of the most beautiful examples is found in the feet of a gecko. A gecko can walk straight up a pane of glass or even hang from a ceiling by a single toe. For a long time, people thought they used suction or some kind of sticky glue. But we now know that they use van der Waals forces. And the bottoms of their feet are covered in millions of tiny hair-like structures called setae. These hairs are so fine that they can get close enough to the atoms of the glass for those fleeting glances, the van der Waals interactions, to take hold. Individually, the hairs do nothing, but the cumulative strength of millions of them is enough to support the gecko's entire weight. It is a literal manifestation of the power of weak forces in the macroscopic world. This knowledge is also the foundation of modern drug design. When scientists try to create a new medicine, they are essentially trying to build a piece of molecular Velcro that will stick to one specific protein in your body and nowhere else. They study the pocket of that protein, its shape, its charges, its water-hating regions, and they design a molecule that fits perfectly. A good drug doesn't just hit a target, it recognizes it through a precise arrangement of hydrogen bonds and ionic interactions. Even the way you perceive the wetness of water is a result of these interactions. Water feels wet because it is constantly forming and breaking hydrogen bonds with the molecules on the surface of your skin. It is reaching out to you. If water couldn't form these non-covalent interactions, it would bead up and roll off you like a dry marble. So, as you rest your thoughts, consider the idea that your very existence is a triumph of the weak. You are not a rigid, unchanging machine. You are a dynamic, flowing masterpiece held together by a trillion gentle handshakes. These forces are the reason you can adapt, the reason you can heal, and the reason you can dream. They are the invisible threads that weave the fabric of your life. Don't worry about the names of the dipoles or the physics of the charges. Just let the logic of it wash over you. The huddling for warmth, the shoving for space, and the gentle embrace of the water that surrounds it all. You are a miracle of molecular cooperation. We have explored the social world of the cell, the Velcro and the shoves that shape the molecules of life within their watery home. We've seen how the hydrophobic effect and the simple attraction of opposites create the complex machines that perform the work of our bodies. But there is one more thing water does that we haven't yet discussed. Sometimes, water doesn't just act as a stage or a social director. Sometimes, it actually breaks apart. When a water molecule loses its composure and splits into pieces, it changes the very nature of the environment. This leads us to the concept of acids and bases. In our next lesson, we are going to explore what happens when water ionizes and how the resulting balance, the pH scale, determines whether life can thrive or whether it will fall apart. Let's hold that thought here, a complete piece of the puzzle. Our next lesson will hand us the adjacent piece, which is the study of the delicate chemical balance of the cellular sea. This is a good place to rest your thoughts for a moment before we continue. If you are still awake, try to visualize those tiny, fleeting handshakes within you, holding everything together with a strength that is as soft as it is certain. You are a wonder of balance and interaction. As you drift deeper into sleep, know that these invisible forces are standing guard over your cells, keeping the dance in motion so that you can rest in peace. When we return, we will look at how life maintains its balance when the water itself begins to change. Sleep well, and I will be here when you are ready to take the next step. Lesson 8. Ionization of water, weak acids, and weak bases. Up until this point in our journey, we have treated water as a stable, supportive stage, the gentle sea that carries the molecules of life. We've admired its stickiness and its ability to act as a universal solvent. But today, we take a closer look at a very rare and very special event. Every now and then, a water molecule does something quite out of character. It falls apart. It is a moment of internal tension where the bonds we've discussed so much finally reach a breaking point, and the molecule splits into two charged pieces. This event, though it happens only to a tiny fraction of molecules at any given time, is the foundation for the entire chemistry of acids and bases. In this lesson, we are going to explore this ionization of water and see how the balance between these pieces creates the environment we call the pH scale. We will look at how weak acids and bases act as the negotiators of the cell, holding on to or releasing their protons in response to the world around them. As we explore this together, try to imagine the cellular sea not just as a static liquid, but as a shifting, breathing balance of charges, where even a slight change in the wind can alter the very rules of life. To understand this lesson, we must look at the internal life of a water molecule, H2O. Usually, the two hydrogens are held firmly by the oxygen atom, in that V shape we've come to know. But remember, oxygen is very greedy for electrons. Sometimes, that pull is so strong that one of the hydrogen atoms is pulled away entirely, but it leaves its electron behind with the oxygen. What remains are two ions, a positively charged hydrogen ion, H+, which is essentially just a lone proton, and a negatively charged hydroxyl ion, OH-. In reality, that lone proton, H+, doesn't like to be alone for long. In the crowded sea of the cell, it almost immediately hitches a ride on another water molecule, creating H3O+, which we call the hydronium ion. For our purposes, though, we can simply think of it as the presence of free protons. This process is called the self-ionization of water. It is an extremely rare event. At any given moment in a glass of pure water, only about 1 in every 555 million molecules is broken apart. It is as if only one person in the entire population of North America decided to step out of their house at the exact same moment. Because this process is a two-way street, a state of equilibrium, the molecules are constantly falling apart and zipping back together. In pure water, the number of H+ ions is always exactly equal to the number of OH- ions. We call this state neutral, but life is rarely neutral. When we add other substances to the water, they can change this balance. If a substance adds more H+ ions to the water, we call it an acid. If it removes H+ ions or adds OH- ions, we call it a base. To measure this balance, we use the pH scale. I want you to take a moment and let the logic of this scale wash over you, because it is a bit unusual. The P in pH stands for the mathematical power, and the H stands for hydrogen. The scale usually runs from 0 to 14. A pH of 7 is neutral. Anything lower than 7 is acidic, and anything higher than 7 is basic or alkaline. The most important thing to remember is that this scale is logarithmic. This means that a change of just 1 unit on the scale is actually a tenfold change in the number of protons. A pH of 5 is 10 times more acidic than a pH of 6, and 100 times more acidic than a pH of 7. It is like the difference between a gentle breeze and a gale force wind. Even a tiny shift in pH represents a massive change in the chemical landscape. Now, as we add layers to this understanding, we must talk about personality. In the world of chemistry, not all acids and bases are created equal. Some are strong, like the hydrochloric acid in your stomach. These are aggressive, and the moment they touch water, they fall apart completely, throwing all their protons into the solution. But in biochemistry, we are much more interested in the weak acids and bases. These are the shy, careful molecules. When a weak acid is placed in water, it only gives up some of its protons. It exists in a state of constant negotiation with the water, constantly letting go of protons and then grabbing them back. We measure this willingness to give using a value called the Ka, or the acid dissociation constant. If an acid has a high Ka, it is more willing to give up its protons. Because these numbers can be very small and difficult to work with, we use the pKa, which is simply the negative log of the Ka. Just like the pH scale, the pKa helps us understand the sweet spot of an acid. If the pH of the environment is exactly equal to the pKa of the acid, then exactly half of the acid molecules have given up their protons, and half are still holding on. This balance is governed by the Bronsted-Lowry definition of acids and bases, which was proposed in 1923. It's a very simple and beautiful way to look at it. An acid is a proton donor, and a base is a proton acceptor. Every acid has a conjugate base, the piece that remains after the proton is given away. They are like partners in a dance. The acid steps forward to offer a proton, and the base steps back to receive it. Take a moment to let that idea find its place. The idea that the strength of an acid is just its willingness to share. In the cell, this is critical because many of our most important molecules, like amino acids and DNA, are themselves weak acids or bases. Their shape and their function depend entirely on whether they are holding on to their protons or not. If the pH of the cell changes, these molecules might gain or lose protons, causing them to change shape, lose their stickiness, or stop working altogether. This is why the study of pH is not just about chemistry. It is about the very survival of the cell's machinery. If your mind is still following along, try visualizing the molecules in your blood right now. They are surrounded by water that is constantly ionizing and recombining. The proteins in your blood are covered in tiny sites that are either holding a proton or looking for one. It is a constant microscopic negotiation that keeps your body in perfect balance. You might wonder how this invisible balance affects your daily life. One of the most striking examples is the gastric acid in your stomach. To digest the proteins in your food, your stomach cells pump out H+ ions until the pH drops to around 1 or 2. This is incredibly acidic, enough to dissolve a piece of metal over time. This high concentration of protons forces the proteins in your food to unfold, making them easier for enzymes to clip into pieces. But notice the cleverness of the design. Your blood, which is just inches away, must stay at a very steady pH of 7.4. If your blood pH dropped to the level of your stomach acid, it would be instantly fatal. Your body has to maintain these two vastly different environments side by side. We can also see this in the food we eat. Have you ever noticed how lemons and vinegar have a sharp, bright taste? That is your tongue detecting the high concentration of protons, the acidity. On the other hand, many medicines are slightly basic, which gives them a bitter taste. Your body is a highly sensitive pH meter, evolved to recognize these chemical signals as signs of energy, spoilage, or danger. Even the way we treat heartburn is a lesson in this chemistry. When you take an antacid, you are essentially swallowing a base, usually something like calcium carbonate. This base acts as a proton sponge, soaking up the excess H+ ions in your esophagus and raising the pH back to a comfortable level. You are performing a chemical neutralization reaction right inside your own chest. So you see, this isn't just theory in a book. It's the principle that explains why your blood is slightly alkaline, why your stomach is acidic, and why a single wrong proton in the wrong place can change the way a protein behaves. It is the subtle, charged landscape that all of life must navigate. We have explored the ionization of water, the rare but powerful moment when the stage itself breaks apart. We've seen how the balance of protons defines the pH scale, and how weak acids and bases act as the negotiators of the cell, carefully managing their protons to maintain their shape and function. But this raises a very important question. If pH is so critical, and if even a small change is so dangerous, how does the body prevent those changes from happening? If you drink a glass of acidic orange juice, why doesn't your blood pH plummet? If you exercise and your muscles produce acid, why don't your cells fall apart? The answer lies in the shock absorbers of the chemical world. In our next lesson, we are going to explore the world of buffering against pH changes in biological systems. We will look at how the body uses its weak acids and bases as a protective shield, soaking up excess protons or releasing them to keep the environment perfectly stable. Let's hold that thought here, a complete piece of the puzzle. Our next lesson will hand us the adjacent piece, which is the study of how life maintains its composure in a world of constant change. This is a good place to rest your thoughts for a moment before we continue. If you are still awake, try to visualize the balance within you, the protons moving back and forth, the water zipping together, and the quiet, steady rhythm of your own internal sea. You are a wonder of equilibrium. As you rest, let the logic of these balances give you a sense of peace. Your body knows how to keep its center, even when the world shifts. Sleep well, and I will be here when you are ready to explore the shields that protect the life within you. Lesson 9, Buffering Against pH, Changes in Biological Systems. In our journey together thus far, we have seen that life is a delicate, shimmering balance of forces. In our last session, we looked at the ionization of water and the profound impact that a few extra protons can have on the environment of the cell. We learned that the pH scale is logarithmic, meaning that a seemingly small shift in number represents a massive tidal wave of chemical change. If the pH of your blood were to shift just a fraction too far in either direction, the very proteins that carry your breath and build your strength would begin to unravel like a frayed tapestry. It seems, at first glance, like a terrifyingly fragile existence. How could something so complex survive in a world filled with acidic lemons, carbonated drinks, and the constant metabolic waste of our own muscles? In this lesson, we explore the answer to that riddle. We are going to look at the shock absorbers of the molecular world, the buffers. These are the quiet, tireless guardians of equilibrium that stand between life and the chaos of drastic chemical shifts. We will explore how these systems act like chemical sponges, soaking up excess acidity or releasing it when needed to keep the environment perfectly steady. As we explore this together, I want you to feel a sense of profound security. You are not a fragile porcelain doll. You are a resilient, self-correcting masterpiece of biological engineering. Take a moment to let that idea find its place. The idea that within you, right now, a silent army of molecules is standing guard on ensuring that the sea of your internal environment remains calm and constant. To understand a buffer, we must look at the partnership between a weak acid and its conjugate base. We spoke in our last lesson about how a weak acid is a negotiator It doesn't give up all its protons at once, but rather exists in a state of balance with its environment. This negotiation is the secret to its protective power. Imagine you have a solution of a weak acid, which we can call HA, and its partner base, which we can call A-. This pair of molecules forms a buffer system. Think of this system not as a static mixture, but as a living, breathing sponge. If we add a strong acid to the system, throwing a handful of angry protons, H+, into the mix, the conjugate base, A-, immediately steps forward, and it catches those extra protons and holds onto them, turning back into the weak acid HA. Because the protons are now bound to the base, they are no longer free to roam the solution and lower the pH. The sponge has soaked them up. Conversely, if we add a strong base, something that tries to steal protons away, the weak acid HA steps forward and releases its own protons to replace the ones that were lost. The sponge has been squeezed, releasing exactly what was needed to maintain the balance. This beautiful process is governed by Le Chatelier's principle, which states that if a system at equilibrium is disturbed, it will shift to counteract that disturbance. It is a natural pushback against change. In the context of biochemistry, we call the range of pH, where a buffer is most effective, the buffering region. This region is usually found within 1 pH unit on either side of the acid's pKa. Within this narrow window, the buffer is at its strongest. It is like a well-tuned spring. It can absorb the shocks of incoming acids or bases without allowing the overall pH to move significantly. If your mind is still following along, try visualizing a titration curve. Imagine we are slowly adding a strong base to a weak acid. At first, the pH rises sharply, but as we enter the buffering region, the curve flattens out. It becomes a gentle horizontal plateau. In this region, even as we add more and more base, the pH barely moves. The buffer is working at its peak, sacrificing its own state to protect the stability of the whole. Don't worry about the mathematics of the curve for a moment. Just visualize that steady, de-level plateau, the safe haven that life carves out for itself in a changing world. To truly understand how we calculate and predict this behavior, we look to a mathematical tool called the Henderson-Hasselbalch equation. It was developed in the early 20th century by two scientists, Lawrence Joseph Henderson and Carl Albert Hasselbalch, who were looking for a way to describe the balance of the blood. The equation is elegant in its simplicity. pH equals pKa plus log open parenthesis, open bracket, a minus close bracket over open bracket, H a close bracket, close parenthesis. This formula tells us that the pH of a buffered solution is determined by two things: the inherent strength of the acid, the pKa, and the ratio of the base to the acid. If the amount of base, A-, is exactly equal to the amount of acid, HA, then the log of that ratio is 0, and the pH is exactly equal to the pKa. This is the midpoint of the buffer, its point of maximum strength. It is the perfect balance where the sponge is exactly half full and half empty, ready to respond to a change in either direction. In our own bodies, the most important buffer system is the bicarbonate buffer system. It is a fascinating multi-layered dance that connects our blood to our breath. The system relies on the balance between carbon dioxide, CO2, water, H2O, carbonic acid, H2CO3, and bicarbonate, HCO3-. The chemical reaction looks like this: CO2 plus H2, O yields H2, CO3 yields H, CO3- and plus H2, CO3 is in equilibrium with H, CO3 minus plus H plus. This system is extraordinary because it is open. If your blood becomes too acidic, if there are too many H plus ions, the reaction shifts to the left. The excess protons combine with bicarbonate to form carbonic acid, which then breaks down into water and CO2. You then simply breathe out that extra CO2. Your lungs are, in a very real sense, a vital part of your blood's buffering system. By breathing faster or slower, you can adjust the amount of CO2 in your system and, by extension, the pH of your entire body. We must also mention the phosphate buffer system, which works primarily inside our cells, and the protein buffer system. Because proteins are covered in amino and carboxyl groups, which we know can act as weak acids and bases, they are incredibly effective buffers. Hemoglobin, the protein that carries oxygen in your red blood cells, is a powerful buffer that helps manage the acidity produced as your tissues work. Historically, the discovery of these systems changed the face of medicine. Before we understood buffering, doctors struggled to explain why certain illnesses led to acidosis, a dangerous drop in blood pH. We now know that during conditions like uncontrolled diabetes, the body produces acidic ketone bodies faster than the buffers can soak them up. This overwhelms the system. The sponge becomes saturated, and the pH begins to fall. Understanding the math of the Henderson-Hasselbalch equation allows modern doctors to precisely calculate how much bicarbonate a patient needs to restore their internal balance. It is a triumph of chemistry applied to the preservation of life. So you see, this isn't just a collection of symbols and equations. It is the principle that allows you to live an active, varied life. When you engage in intense exercise, your muscles produce lactic acid without your buffering systems. Now, that acid would instantly shut down your metabolic pathways. Instead, your bicarbonate and protein buffers go to work. You begin to breathe more deeply and quickly, exhaling the CO2 that represents the acid you've neutralized. You are performing high-level biochemistry with every labored breath. This also explains why we can enjoy acidic foods like citrus fruits or vinegary dressings. While these foods are acidic on your tongue, by the time they are absorbed into your blood, your buffering systems have already prepared for their arrival. They maintain that steady pH of 7.4 with a precision that is nothing short of miraculous. Think of your buffering systems as the wisdom of your body. They represent a deep ancestral knowledge of how to maintain peace in a world of conflict. They remind us that stability is not the absence of change, but the ability to respond to change with grace and balance. As you rest tonight, let that thought comfort you. Your body is not a static object. It is a dynamic, self-balancing system that is constantly working to keep you safe, even while you dream. Don't worry about the details of the pKa values or the log tables. Just let the logic of it wash over you. You are protected. You have shock absorbers built into your very atoms. You are a masterpiece of equilibrium, perfectly buffered against the storms of the world. We have explored the guardians of the cellular sea, the buffers that protect us from the volatility of pH. We've seen how weak acids and bases work together as a chemical sponge, and how our very breath helps maintain the balance of our blood. This concludes our study of the environment in which life exists, the water, the ions, and the protective shields. But life is more than just a stable environment. Life is movement. Life is growth. Life is the constant transformation of one thing into another. All of this requires something more than just a stage and a buffer. It requires energy. How do we harness the energy from the sun or our food to power the complex machines of the cell? How do we know which reactions will happen and which ones won't? Let's hold that thought here. A complete piece of the puzzle. Having now grasped the stability of the environment, we are perfectly prepared to appreciate the energy that drives the activity within it. Our next lesson will hand us the adjacent piece, which is a study of thermodynamics and the laws of energy. And we will move from the where of life to the how of life's power. This is a good place to rest your thoughts for a moment before we continue. If your mind is still following along, try to visualize that steady titration plateau, a place of calm and balance. You are well guarded. You are in equilibrium. And when we return, we will begin our study of the fire that drives the dance. Sleep well, and I will be here when you are ready to take the next step. Lesson 10. Thermodynamics and the laws of energy. In our journey together thus far, we have explored the stage of life, the water, the elements, and the delicate chemical balances that keep the cellular sea calm. We have admired the architecture and the tools. But now, we must ask the most fundamental question of all. What drives the movement? Why does a seed push through the dark earth to find the sun? Why does a heart beat with such rhythmic persistence? The answer lies in the profound and inescapable laws of energy. In this lesson, we are going to explore thermodynamics. I know the word can sound cold or mechanical, reminiscent of steam engines and heavy machinery, but in biochemistry, thermodynamics is the very poetry of existence. It is the study of how energy flows, transforms, and eventually dictates what is possible and what is forbidden in the living world. We are going to look at the two great laws that govern every spark of life, from the smallest bacterium to the vastness of a redwood forest. As we explore this together, I want you to think of energy not as an abstract number in a textbook, but as the currency of life. Every breath you take is a transaction, a careful exchange with the universe. Take a moment to let that idea find its place. The idea that you are a localized whirlpool of energy, spinning in a vast cosmic ocean. To understand the energy of life, we must first define our boundaries. In thermodynamics, we talk about the system and the surroundings. The system is whatever we are focusing on, perhaps a single cell, or even a single enzyme within that cell. The surroundings are everything else in the universe. Together, they form the universe. In biochemistry, we are almost always dealing with an open system. This is a beautiful concept. A closed system is like a sealed glass jar where nothing can get in or out. But life is an open door. We are constantly exchanging matter and energy with our surroundings. We take in food and oxygen, and we release heat and waste. This openness is the only reason we can stay alive. This brings us to the first law of thermodynamics, often called the law of conservation of energy. It states, quite simply, that energy cannot be created or destroyed. It can only be transformed from one form to another. Think of a waterfall. The water at the top has potential energy because of its height. As it falls, that energy transforms into kinetic energy, the energy of motion. If that water hits a turbine, the kinetic energy becomes electrical energy. In your body, the process is just as magical. You take the chemical energy stored in the bonds of a sandwich and transform it into the kinetic energy of your walk, the electrical energy of your thoughts, and the thermal energy that keeps you warm. The total amount of energy in the universe remains constant, but it is always changing clothes, moving from one form to another to do the work of life. However, the first law doesn't tell the whole story. It tells us that energy is conserved, but it doesn't tell us which way the energy wants to go. For that, we need the second law of thermodynamics. This is perhaps the most profound law in all of science. It introduces us to the concept of entropy, which we represent with the symbol S. Entropy is often described as disorder or randomness, but I want you to think of it as the universe's tendency to spread energy out. Imagine you have a perfectly ordered deck of cards, sorted by suit and number. If you throw those cards into the air, they will land in a messy, disordered pile. This is the natural direction of the universe. It is very easy to go from order to disorder, but it requires a great deal of work to go from disorder to order. The second law states that in every energy transformation, the total entropy of the universe must increase. Some energy is always lost to the surroundings as heat, a disorganized form of energy that can no longer be used to do work. Heat is the tax that the universe collects on every transaction. This presents a fascinating puzzle for biochemistry. If the universe always tends toward disorder, how can life be so incredibly organized? Your cells are the opposite of a messy pile of cards. They are masterpieces of exquisite, intricate order. Does life break the second law? Not at all. We maintain our internal order by creating an even greater amount of disorder in our surroundings. We take organized, low-entropy molecules like glucose and break them down into simple, high-entropy molecules like carbon dioxide and water while releasing heat. We are like a person who keeps their own room spotless by throwing all their trash out into the street. We stay ordered by exporting entropy to the rest of the universe. To truly grasp the how of this energy flow, we must look at enthalpy, which we represent with the symbol H. As if entropy is about the spread of energy, enthalpy is about the content of energy, specifically the heat content. In biochemistry, enthalpy usually refers to the energy stored in chemical bonds. When a reaction releases heat into the surroundings, we call it exothermic or exergonic in terms of total energy. Think of a crackling fire. It is releasing enthalpy as heat. When a reaction absorbs heat, we call it endothermic. In the late 19th century, scientists like Rudolf Clausius and Lord Kelvin began to realize that life was essentially an energy transducer. We aren't just consumers of energy. We are sophisticated machines that capture energy in one form and precisely channel it into another. We must consider the total internal energy, E, of a system, but in the soft, wet environment of a cell, where pressure and volume don't change much, we focus on the relationship between enthalpy, entropy, and temperature, T. If your mind is still following along, try to visualize the cost of living. Every time an enzyme builds a strand of DNA, it is fighting a battle against the second law. It is trying to create order, low entropy, out of a chaotic soup of building blocks. To win this battle, the enzyme must pay for that order using energy, usually from ATP. And in doing so, it releases heat. This is why you feel warm when you exercise. Your muscles are working hard to create the ordered state of contraction, and they are paying a heavy entropy tax in the form of heat. Don't worry about the complex calculations or the history of steam engines. Just let the logic of it wash over you. Life is a constant, an uphill struggle against the natural tendency of things to fall apart. We are the exceptions to the rule of chaos, but we are exceptions that prove the rule. We are only able to exist because we are so efficient at processing energy and passing the disorder along to the world around us. This is the fundamental molecular logic of life. We are energy processing islands in a sea of increasing entropy. So you see, thermodynamics isn't just a theory for physicists. It's the reason you have to eat breakfast. Your body needs a constant stream of low-entropy order in the form of food to counteract the relentless disorder that is trying to pull your cells apart. If you stop eating, your body eventually loses the battle against entropy, and the organized structure of your life dissolves back into the simple, disorganized atoms of the earth. This is what we call equilibrium. In thermodynamics, true equilibrium for a living organism is quite literally death. To be alive is to be held in a steady state that is far away from equilibrium. Think of a fountain. As long as the pump is running, the water stays in a beautiful, organized shape, a steady state. But the moment you turn off the power, the water falls into a flat, characterless pool, equilibrium. You are that fountain. Your metabolism is the pump, and the food you eat is the power. This also explains the efficiency of biological systems. While a car engine might only convert about 20% of its fuel's energy into movement, wasting the rest as heat, your biological engines are often much more efficient. However, they are still bound by the second law. No process is 100% efficient, and there is always a cost. This is why even the most perfect diet and exercise routine cannot stop the gradual messiness that accumulates in our cells over decades, what we perceive as the process of aging. It is the slow, inevitable accumulation of entropy that our repair mechanisms eventually can't keep up with. This is a good place to rest your thoughts for a moment. If your mind is still awake, try to visualize yourself as that fountain, a beautiful, organized shape made of flowing energy. You are a temporary triumph of order over chaos. It is a wondrous thing to be an island of complexity in a universe that is slowly spreading out and cooling down. We have explored the two great laws of the energy economy: the conservation of energy and the inevitable increase of entropy. We've seen how life maintains its internal mansion by exporting its mess to the backyard of the universe. We've looked at the what and the why of energy flow. But there is still one vital question. How do we know if a specific reaction will actually happen? If we have a certain amount of enthalpy and a certain change in entropy, how do we calculate the bottom line? How do we know if a reaction has enough profit to proceed on its own? Let's hold that thought here, a complete piece of the puzzle. Having now grasped the laws of energy, we are perfectly prepared to appreciate the tool we use to measure that profit. Our next lesson will hand us the adjacent piece, which is a study of free energy and chemical equilibrium. We will look at the work of Josiah Willard Gibbs and the single most important number in biochemistry, delta G. This is a good place to rest your thoughts for a moment before we continue. Let the logic of the flowing energy stay with you. You are a masterpiece of thermodynamics, a quiet flame burning steadily in the night. Sleep well, and I will be here when you are ready to take the next step. Lesson 11. Free energy and chemical equilibrium. In our previous session, we walked through the hallowed halls of thermodynamics, learning the two great laws that govern the universe's energy economy. We saw that energy is always conserved, but that it has an inescapable tendency to spread out and become disorganized, a process we call entropy. But for those of us interested in the living world, these laws leave us with a burning question. If some energy is always lost to the environment as heat and disorder, how do we know exactly how much useful energy is left to actually do the work of a cell? How does a cell know if a specific chemical reaction, say, the building of a protein or the firing of a neuron, is even possible in the first place? In this lesson, we are going to explore the concept of free energy and its relationship with chemical equilibrium. We are going to meet the tool that allows us to calculate the bottom line of any biological transaction. We will move beyond the raw heat and the raw messiness to find the single most important number in biochemistry, the Gibbs free energy. As we take this next step together, try to imagine that we are looking at the available cash in the cell's bank account. It is the energy that is free and ready to be spent on the grand project of staying alive. Take a moment to let that idea find its place. The transition from the total energy of the universe to the specific, spendable energy of you. To understand free energy, we must look to the work of a quiet, brilliant man named Josiah Willard Gibbs. In the late 1800s, he developed a single equation that combined the heat of a reaction and the disorder of a reaction into one measurable value. We represent this as G, or Gibbs free energy. When a chemical reaction occurs, we look at the change in this value, which we call delta G, or delta G. The formula is a beautiful balance of the forces we have already studied. Delta G equals delta H minus T delta S. In this elegant expression, delta H is the change in enthalpy, the total heat content. T is the absolute temperature, and delta S is the change in entropy, the measure of disorder. What this equation is essentially telling us is that the useful energy left over after a reaction, delta G, is the total energy released as heat, delta H, minus the tax that must be paid to the universe's disorder, T delta S. Now, I want you to visualize this delta G value as a compass. It tells us the direction in which a reaction will naturally flow. If a reaction has a negative delta G less than 0, it means the system is losing free energy. It is releasing energy that can be used to do work. We call such a reaction exergonic. Think of a ball sitting at the top of a steep hill. If you give it a tiny nudge, it will roll down on its own. It is a spontaneous process. In the world of the cell, exergonic reactions are the ones that power our lives. They are the downhill slides that release the energy we need. Conversely, if a reaction has a positive delta G greater than 0, it is endergonic. This means the reaction requires an input of energy to proceed. It is like trying to push that same ball back up the hill. It will not happen on its own. You must pay the energy cost to make it go. Most of the creative work of life, building new DNA, constructing cell walls or moving muscles, is endergonic. These reactions are uphill battles that require a constant investment of energy. Then, we reach a very special state, chemical equilibrium. This occurs when delta G is exactly 0. At this point, the forward and backward reactions are happening at the exact same rate. There is no longer any net change, and no energy can be extracted to do work. If your mind is still following along, try visualizing a perfectly level seesaw. Both sides are balanced, and nothing is moving. While equilibrium is a goal for many things in physics, in biochemistry, it is a state to be avoided at all costs. As we've mentioned before, a cell at true chemical equilibrium is a cell that can no longer do work. It is a dead end. To be alive is to be held in a state of disequilibrium, constantly rolling downhill through one reaction and being pushed back up through another. If your mind is wandering, try visualizing the water in a river. As long as the water is flowing from a high place to a low place, it can turn a water wheel and grind grain. That flow is like a reaction with a negative delta G. But if the water reaches a perfectly flat, still lake, the wheel stops turning. That stillness is equilibrium. Life is the river, never the lake. As we add layers to this understanding, we must distinguish between the delta G happening in your cells right now and what we call the standard free energy change, or delta G naught. Because the spendable energy of a reaction depends on the concentration of the chemicals, the temperature, and the pressure, scientists needed a baseline for comparison. They created the standard state, a set of controlled conditions where everything is at a specific concentration and temperature. This delta G naught is like the list price of a reaction in a catalog. It tells us how much energy the reaction would release or require under those perfect conditions. However, the actual delta G in your body is often very different from the list price. We calculate it using the equation. Delta G equals delta G naught plus RT natural log Q. Here, R is the gas constant, T is the temperature, and Q is the ratio of products to reactants currently in the cell. This means that a cell can actually drive a reaction that might seem impossible by simply changing the concentrations. If a cell quickly uses up the products of a reaction, it keeps the ratio low, which can turn a positive delta G into a negative one. It is a clever way for life to manipulate the laws of chemistry to suit its needs. We also see a profound relationship between free energy and the equilibrium constant, K sub EQ. The more negative the delta G naught is, the more the reaction wants to move toward products, and the larger the equilibrium constant will be. The formula is delta G naught equals minus RT natural log K sub EQ. This tells us that the potential energy of a reaction is directly tied to where the balance point eventually lies. But how does a cell perform those uphill endergonic reactions that have a positive delta G? It uses a beautiful strategy called reaction coupling. This is perhaps the most important concept in all of metabolism. The cell takes an expensive reaction, one with a positive delta G, and physically links it to a very cheap energy-releasing reaction, one with a large negative delta G. As long as the total sum of the two reactions has a negative delta G, the whole process will proceed. The currency for this coupling is almost always ATP, or adenosine triphosphate. The breaking of a phosphate bond in ATP is a highly exergonic downhill slide. By coupling the downhill break of ATP to the uphill build of a protein, the cell makes the impossible possible. Think of it like a professional athlete. They have the expensive task of training all day, endergonic, but they pay for it by using the income from their sponsorships, exergonic. As long as they earn more than they spend, they can keep training. This is how you are built. You are a series of expensive, beautiful reactions paid for by the constant burning of the fuels you eat. Take a moment to let that idea find its place. The idea of life as a series of coupled gears, one turning the other, ensuring that the work of the mansion never stops. You are a masterpiece of energy management, a system that knows exactly how to spend its cash to keep the lights on and the structure sound. You might wonder why we spend so much time on the math and the theory of free energy while you are trying to rest. It is because this is the principle that explains why you feel tired when you are hungry. When your blood sugar is low, your cells have less raw material to drive those exergonic reactions. Your supply of energy cache, the ATP, starts to dwindle. Because you can no longer pay for the uphill reactions of movement and thought, your body slows down to conserve what little free energy it has left. This also explains the logic of metabolic pathways. Your body doesn't just break down a sugar cube in one giant explosion. If it did, most of that energy would be lost as heat, enthalpy, which could actually damage your cells. Instead, your body breaks it down in dozens of tiny, incremental steps. Each step has a small, manageable delta G. This allows the cell to capture that energy efficiently, using it to build ATP like a person putting pennies into a jar. By the time the sugar is completely broken down, the cell has collected a significant amount of spendable energy without ever causing a dangerous spike in temperature. Even the process of folding a protein is a lesson in free energy. As we saw in our lesson on the hydrophobic effect, the water shoves the non-polar parts of a protein together. This increase in the entropy of the water, delta S, is so large that it makes the overall delta G of the folding process negative. The protein folds spontaneously because the universe wants the water to be more disordered. You are shaped by the universe's desire for messiness. So you see, delta G is not just a letter in an equation. It is the invisible hand that guides every process in your body. It is the reason your heart knows how to beat and why your brain can process the words I am saying. It is the measure of your vitality, the ledger of your existence. Don't worry about the specific values of R or the natural logs. Just let the logic of it wash over you. You are a system that is perfectly tuned to harvest, store, and spend the energy of the universe to maintain the miracle of you. We have explored the bottom line of biochemistry, the free energy that tells us if a reaction is possible. We've seen how life couples downhill and uphill reactions to build its complex structures, and how it avoids the stillness of equilibrium. We now know what can happen. But there is one final piece to this puzzle. Just because a reaction has a negative delta G, just because it can happen, doesn't mean it will happen quickly. A diamond has a negative delta G to turn into graphite, but that process takes billions of years. Life cannot wait billions of years for its chemistry to happen. It needs its reactions to happen in the blink of an eye. Let's hold that thought here. A complete piece of the puzzle. Having now grasped the possibility of reactions, we are perfectly prepared to appreciate the speed of reactions. Our next lesson will hand us the adjacent piece, which is a study of reaction kinetics and catalysis. We will move from thermodynamics to kinetics, meeting the master accelerators of life, the enzymes. This is a good place to rest your thoughts for a moment before we continue. If your mind is still following along, Try to visualize the downhill flow of energy within you. A steady, quiet stream that powers everything you are. You are well funded by the laws of nature. You are in a state of beautiful disequilibrium. And when we return, we will see how life speeds up the dance to the pace of a heartbeat. Sleep well, and I will be here when you are ready to take the next step. Lesson 12. Reaction, Kinetics, and Catalysis. In our last few sessions, we have mastered the laws of the possible. We explored thermodynamics and the concept of free energy, learning that a negative delta G is the universe's way of giving a reaction a green light. But as we transition into this 12th lesson, we must confront a startling truth. In the world of biochemistry, a green light does not guarantee a high speed. There are reactions happening within you right now that are thermodynamically downhill, meaning they release energy and want to happen, yet, if left to their own devices, they would take centuries to complete. A diamond, for example, is thermodynamically unstable relative to graphite. It wants to turn into pencil lead, but that process takes billions of years. Life, as you well know, cannot wait billions of years. Life is a race against time. To be alive is to perform millions of chemical reactions every second with the precision of a master watchmaker. In this lesson, we move from the if of thermodynamics to the when of reaction kinetics. We are going to explore the energy barriers that stand in the way of change and meet the extraordinary molecular machines called enzymes that act as the master accelerators of existence. Take a moment to let that idea find its place. The transition from the map of the journey to the speed of the engine, graph of potential energy and reaction coordinate. To understand why some reactions are slow, we must visualize the energy barrier. Imagine you are standing at the top of a beautiful, snow-covered hill with a sled. According to thermodynamics, the slide down is spontaneous. It has a negative delta G. You are at a higher energy state at the top than you will be at the bottom. However, imagine that right in front of your sled, there is a small, stubborn mound of snow. Even though you are destined to go down, you cannot start until you push your sled over that initial hump. In chemistry, we call this hump the activation energy, often represented as E sub A or delta G dagger. Every chemical reaction, even the most energetic one, requires an initial investment of energy to get started. This is because for a reaction to occur, bonds must be stretched, twisted, or broken before new ones can form. We call the moment of maximum tension, the very top of that snow mound, the transition state. It is a fleeting high-energy arrangement where the old bonds are half broken and the new bonds are half formed. It is the most unstable moment in a molecule's life. If a molecule doesn't have enough kinetic energy to reach this summit, the reaction simply won't happen, no matter how much energy is waiting to be released on the other side. This is where kinetics comes in. Kinetics is the study of the rate or speed of these reactions. In a simple world, you could speed up a reaction by adding heat. Heat makes molecules move faster, causing them to collide with more force, and a higher chance of overcoming that activation energy barrier. But your body cannot simply turn up the heat. If your internal temperature rose high enough to make your metabolic reactions happen fast enough on their own, your delicate proteins would cook and coagulate like an egg on a frying pan. Life needed a different strategy. Life chose catalysis. A catalyst is a substance that speeds up a chemical reaction without being consumed by it. It doesn't change the starting point, and it doesn't change the finish line, meaning it has no effect on the thermodynamics or the delta G. Instead, it provides an alternative route, a mountain pass, that allows the reaction to proceed with a much lower activation energy. In the biological world, these catalysts are almost always proteins called enzymes. An enzyme is a master of the transition state. It doesn't just sit there, it actively reaches out and grabs the reacting molecules, which we call substrates, and coaxes them into the shape of the transition state. By stabilizing that fleeting, unstable moment, the enzyme lowers the height of the snow mound, allowing trillions of molecules to slide downhill every second at normal body temperature. Take a moment to let that logic wash over you. The idea that life isn't about brute force or high heat, but about the elegant lowering of barriers. If your mind is still following along, try to visualize an enzyme as a specialized cradle, perfectly shaped to hold a molecule in the exact moment of its transformation. As we go deeper into the mystery of how enzymes work, we must look at the active site. This is a small specialized pocket, or groove, on the surface of the enzyme. It is lined with specific amino acids and functional groups, the tools we studied in Lesson 5, that are arranged with subatomic precision. For a long time, we used the lock and key model to describe this, suggesting that the substrate fits into the active site like a key into a lock. However, we now prefer a more dynamic description called the induced fit model. I want you to imagine a glove. When your hand is not in it, the glove has a general shape. But as you slide your hand inside, the glove stretches and contours itself to fit your fingers perfectly. An enzyme does the same. When the substrate binds, the enzyme actually changes its shape, wrapping around the substrate in a molecular hug. This change in shape does two vital things. First, it pushes the reacting atoms closer together in the perfect orientation. Second, it puts physical or electrical strain on the bonds that need to be broken. The energy that allows the enzyme to do this is called binding energy. When the enzyme and substrate touch, and they form many of those weak non-covalent interactions we studied in Lesson 7, hydrogen bonds, ionic interactions, and van der Waals forces. Even though they are weak individually, the sum of these interactions releases a small burst of energy. This binding energy is the payment that the enzyme uses to lower the activation energy barrier. It is a beautiful, self-sustaining system. The act of binding provides the energy needed to facilitate the reaction. Historically, our understanding of enzymes began in the 19th century with the study of fermentation. In fact, the word enzyme comes from the Greek enzyme, meaning in yeast. Scientists like Edward Buchner proved that you didn't need a whole living cell to perform these reactions. You just needed the specific proteins within them. Later, in 1913, Leonor Michaelis and Maud Menton developed the mathematical framework that allows us to calculate how fast an enzyme works. They gave us the Michaelis-Menten equation, which describes the speed limit of an enzyme. Every enzyme has a maximum velocity, Vmax, a point where all its active sites are full, and it simply cannot work any faster. We must also consider enzyme inhibition. Sometimes the cell needs to slow down or stop a reaction. It does this by using inhibitors, molecules that bind to the enzyme and prevent it from doing its job. A competitive inhibitor is like a piece of tape stuck over a keyhole. It blocks the active site so the substrate can't get in. An allosteric inhibitor is more subtle. It binds to a different part of the enzyme and causes the whole structure to shift, distorting the active site so the key no longer fits. This ability to turn enzymes on and off is how your body regulates its metabolism, ensuring that you don't produce more energy or building blocks than you actually need. Don't worry about the complex math of the Km values or the reciprocal plots tonight. Just let the logic of it find its place. Life is a choreographed race, and enzymes are the directors, deciding which reactions happen, how fast they go, and when they should stop. They are the reason you can respond to the world in real time. So you see, this isn't just a theory in a book. It is the principle that explains why you can breathe, move, and think. Consider the enzyme carbonic anhydrase. It is one of the fastest enzymes known to man. Its job is to turn carbon dioxide into bicarbonate so it can be carried in your blood, the very buffer system we studied in Lesson 9. Without this enzyme, that reaction would happen so slowly that CO2 would build up in your tissues and you would suffocate in minutes. Carbonic anhydrase speeds up the reaction by a factor of 10 million. It turns a century-long process into a millisecond-long event. This knowledge is also the foundation of most modern medicine. When you take a common pain reliever like aspirin, you are using an enzyme inhibitor. Aspirin travels through your blood and binds to an enzyme called COX2, which is responsible for making the chemicals that cause inflammation and pain. By clogging that enzyme's active site, aspirin stops the production of pain signals at the source. Similarly, many life-saving drugs for high blood pressure or viral infections work by precisely targeting and disabling a single specific enzyme. We can even see this in our diet. People who are lactose intolerant are simply missing a specific enzyme called lactase. Without that molecular cradle to help break the bond in milk sugar, the reaction happens too slowly, or not at all, leading to digestive distress. In this case, the thermodynamics are perfectly fine. The body wants to break down the sugar, but the kinetics are missing the necessary accelerator. If your mind is still following along, try visualizing the millions of enzymes inside you right now. Each one a tiny, tireless worker, catching substrates, hugging them into the transition state, and releasing the products. They are the heartbeat of your chemistry. They are the reason that a simple sandwich can be turned into the energy of a dream. Take a moment to rest your thoughts here. You are not just a collection of chemicals. You are a high-speed, perfectly timed symphony of catalysis. Every second of your life is made possible by these proteins that refuse to let the energy barriers of the universe stand in your way. We have explored the speed of life, the reaction kinetics that allow us to overcome the barriers of activation energy. We've seen how enzymes act as master accelerators, using binding energy to stabilize the transition state and coax molecules into new forms. We've looked at the active site, the induced fit, and the way life regulates its own pace. But as we reach the end of this particular chapter, we must take one final step back. We have looked at the parts, the bonds, and the energy, the water, and the speed. We have seen all the individual players in the drama of biochemistry. Now, we must look at the script. How do all these diverse pieces, the atoms, the delta G, the pH, and the enzymes, integrate into a single, cohesive, and logical system? What is the overarching molecular logic that allows inanimate matter to become a living, breathing being? Let's hold that thought here, a complete piece of the puzzle. Having now grasped the pace of the dance, we are perfectly prepared to appreciate the choreography of the whole. Our final lesson will hand us the adjacent piece, which is a study of the molecular logic of life. We will bring all these foundations together to see the grand synthesis of what it means to be alive. This is a good place to rest your thoughts for a moment before we continue. If you are still awake, try to visualize those tiny, efficient cradles within you, speeding up the world so that you can exist in peace. You are well-timed. You are perfectly catalyzed. And when we return, we will look at the grand logic that ties it all together. Sleep well, and I will be here when you are ready to take the final step in our journey. Lesson 13. The molecular logic of life. We have reached a truly significant moment in our shared journey. Over our past 12 lessons, we have meticulously gathered the pieces of a vast and intricate puzzle. We have looked at the humble atoms, carbon, hydrogen, oxygen, and nitrogen, that serve as the alphabet of existence. We have explored the invisible handshakes of chemical bonds, the elegant three-dimensional geometry of molecules, and the functional groups that act as the tools of the trade. We have plunged into the supportive sea of water, learned the strict energy laws of thermodynamics, and marveled at the high-speed choreography of enzymes. But now, we must step back from the individual gears and the specific reactions. We must look at the grand architecture of the whole. In this final lesson, we explore the molecular logic of life. This is the study of how inanimate, non-living matter organizes itself into something that can breathe, react, grow, and eventually wonder about its own origin. It is the script that directs the play. As we explore this together, I want you to feel the profound sense of unity and purpose that biochemistry reveals. We are moving from the how of individual parts to the why of the entire system. Take a deep, slow breath and let the complexity of the previous lessons settle into a single, unified picture. We are looking at the logic that turns chemistry into a biography. To understand the molecular logic of life, we must first address a beautiful paradox. If you were to look at any single molecule within your body, a strand of DNA, a single protein, or a molecule of ATP, none of those things, in isolation, is alive. They follow the same laws of physics and chemistry that govern a stone or a gust of wind. Yet, when they are brought together in a specific, highly organized arrangement, life emerges. This is the concept of emergence. Life is not a thing you can point to, it is a collective property of a system. The molecular logic of life is the set of ground rules that allows this system to maintain itself against the relentless pull of entropy. The first rule of this logic is the transformation of energy and matter. As we learned in our thermodynamics session, life is an open system. The logic of life dictates that an organism must constantly take in high-quality energy and organized matter from its surroundings. We do not create life. We channel the energy of the universe through our metabolic pathways. Think of a fountain again. The shape of the water is constant, but the individual droplets are always moving, always being replaced. The logic of life is the pump and the nozzle that keeps the fountain's shape steady. This constant flow, this metabolic flux, is what allows us to stay in a state far from equilibrium. We are not static statues. We are a steady, purposeful flow of atoms. The second rule of this logic is self-replication and information flow. For a system to be considered alive, it must have a way to store the instructions for its own construction and pass those instructions to the next generation. This is the logic of the central dogma. DNA makes RNA, and RNA makes protein. I want you to visualize your DNA not just as a molecule, but as a vast ancient library. The logic of life ensures that the books in this library are copied with incredible precision, but also with just enough room for the occasional typo, a mutation that allows for the diversity and adaptation of evolution. Without this information flow, the organized state of the cell would dissolve the moment the individual molecules wore out. Information is the memory of life, ensuring that the architecture of the mansion remains long after the original bricks have been replaced. The third rule is autoregulation and homeostasis. The logic of life is a self-correcting logic. Because the environment is always changing, the temperature shifts, the food supply fluctuates, the pH wavers. The system must be able to sense these changes and respond. This is achieved through thousands of interlocking feedback loops. When you have enough of a certain product, the product itself reaches back and turns off the enzyme that makes it. It is a system of exquisite sensitivity and balance. Think of it as a master thermostat that doesn't just control heat, but controls every single chemical concentration within your cells. This wisdom of the body ensures that despite the chaos of the outside world, the internal environment remains a calm, predictable sanctuary. Take a moment to let those three pillars find their place: the flow of energy, the storage of information, and the constant self-correcting balance. If your mind is still following along, try to visualize yourself as a localized triumph of order, a system that knows itself, maintains itself, and remembers itself through the language of molecules. As we add layers to this grand synthesis, and we must acknowledge the pioneers who first glimpsed this logic. In the mid-20th century, scientists like Albert Leninger began to articulate these principles in a way that bridged the gap between the wet chemistry of the lab and the living reality of the organism. They realized that there is a profound unity of life. When we look at the molecular logic of a tiny E. coli bacterium and compare it to the logic of a human neuron, the similarities are staggering. They use the same genetic code, the same 20 amino acids, and the same ATP energy currency. This tells us that the logic we are studying is not just a collection of random coincidences. It is a highly refined, ancient operating system that has been perfected over billions of years. One of the most profound layers of this logic is the structure-function relationship. In the world of human engineering, we might build a tool to perform a task. In the world of biochemistry, the tool is the task. The shape of a protein is its function. The logic of life dictates that every molecular shape is purposeful. A protein that is shaped like a channel will let things through. A protein shaped like a hook will catch onto its neighbors. This is a level of integration that exceeds anything we can build. Every click and vibration of a molecule is a part of the calculation of being alive. We must also consider the dynamic steady state. We often use the word balance to describe health, but in biochemistry, balance or equilibrium is a dead end. The molecular logic of life is about maintaining a steady state that is purposefully unbalanced. We keep the concentration of certain ions high on one side of a membrane and low on the other, creating a chemical dam that can be released at any moment to send a signal or move a muscle. We are like a cyclist who stays upright, not by being perfectly still, but by constantly moving forward. The moment the movement stops, the balance is lost. This dynamic nature is what gives life its responsiveness and its vibrancy. If you find yourself drifting, try to visualize the millions of feedback loops in your body right now. They are like a vast, silent orchestra. There is no conductor in the center. Instead, every instrument listens to every other instrument, adjusting its own volume and tempo to maintain the harmony. The music produced by this orchestra is your consciousness, your movement, and your rest. It is a logic of cooperation and coordination that happens entirely without thought, yet is more complex than any thought we can conceive. Don't worry about the specific names of the pathways or the details of the genetic code tonight. Just let the beauty of the integration wash over you. You are a system of profound, ancient intelligence. Every atom in you is part of a conversation that has been going on since the first cell appeared on this planet. You are the current high water mark of a 4 billion-year-old logic. So you see, the molecular logic of life is the ultimate synthesis. It explains why we study biochemistry in the first place. This knowledge is the foundation of modern medicine. We no longer view disease as a mysterious curse. We view it as a logic error, a break in a feedback loop, a mutation in the information store, or a blockage in the energy flow. When we develop a new therapy, we are essentially debugging the system, providing a molecular patch to restore the harmony of the steady state. This logic also connects us to the environment. Because we share the same molecular alphabet as every other living thing, we are part of a global web of energy and matter transformation. The logic that keeps you alive is the same logic that keeps the forest breathing and the oceans teeming with life. We are not on the earth, we are of the earth, a part of its total chemical inventory. When we understand our own biochemistry, we gain a deeper respect for the fragility and the resilience of the entire planet. Furthermore, this logic gives us a framework for the future. As we move into the era of synthetic biology and personalized medicine, we are learning to write in the language of life. We are learning to design new enzymes, to correct genetic blueprints, and to optimize metabolic pathways. By understanding the rules of the game, the molecular logic, we gain the ability to heal what was once unhealable and to protect the diversity of life in a changing world. Take a moment to let that find its place. The idea that you are a part of a much larger, incredibly ancient, and beautifully logical whole. This isn't just a science. It is a perspective that changes how you see the world. Every person you meet, every animal you see, and even the tiny microbes you cannot see are all variations on this same magnificent theme. We are all bound by the same chemical foundations. We have come to the end of our foundational course. We have journeyed from the lonely atom to the complex, logical system of the living cell. You have been a patient and thoughtful companion on this intellectual journey. If your eyes are closed and you are on the threshold of sleep, know that the molecular logic we have discussed is now standing guard over you. While you rest, your enzymes are still working, Your DNA is being protected, and your steady state is being maintained with a quiet, tireless devotion. You are a wonder of chemical coordination. You are a masterpiece of thermodynamics and a triumph of molecular information. As the music of these thoughts fades away, let the sense of security and wonder wrap around you like a warm blanket. You are well made. You are a logical, beautiful, and vital part of the universe. There is no more for you to do tonight. The logic is in motion, and it will carry you safely through the night and into the morning. Rest now, in the quiet sanctuary of your own biology. Sleep well. Sleep deeply. The journey for tonight is complete.